Gastric neuromuscular function and neuromuscular disorders. Kenneth L. Cook 50. Gastric neuromuscular function refers to the contractions, relaxations, and peristaltic activities of the stomach. The three major neuromuscular activities of the stomach are: 1. Receptive relaxation of the fundus, 2. Recurrent peristaltic waves of the corpus and antrum, and 3. Antral peristalsis with antropyloroduodenal coordination. These major neuromuscular activities of the stomach accomplish three key functions: 1. To receive the ingested food that we eat, receptive relaxation; 2. To mill, triturate, the ingested foodstuffs into a nutrient suspension termed chyme, and 3. To empty the chyme from the stomach through the pyloric sphincter into the duodenum in a highly regulated fashion. This sophisticated process is necessary to maximize further digestion and absorption of the nutrients in the small intestine. Neuromuscular dysfunction of the stomach results in nausea, early satiety, vomiting of chewed food, and dysregulation of the gastric emptying of solid and liquid nutrients. These critical gastric neuromuscular activities and related functions are complexly modulated by the CNS, parasympathetic nervous system, PNS, and sympathetic nervous system, SNS. The interactions of the CNS and the activity of the enteric nervous system, ENS, the interstitial cells of Kagel, ICCs, that regulate the frequency of smooth muscle contractions and organize peristaltic waves, and the host of neurotransmitters that ultimately regulate the contraction and relaxation of gastric smooth muscle. Electrophysiologic basis for normal gastric neuromuscular function: extracellular slow waves and plateau and action potentials. The stomach is a sophisticated sphere of smooth muscle organized into circular, longitudinal, and oblique muscle layers. Gastric myoelectrical activity, GMA, termed slow waves or pace setter potentials, regulate, control, and pace gastric smooth muscle contractions. 1,2 In the normal human stomach, the slow waves occur at approximately 3 cycles per minute, CPM, or between 2.5 and 3.7 CPM. 3,4 From the pacemaker region on the greater curvature of the stomach, between the fundus and the proximal corpus, slow waves propagate distally and circumferentially toward the pylorus every 20 seconds, with highest amplitude and velocity, approximately 7 mm/sec, in the distal 2 to 4 cm of antrum, Fig 50.1.5. The gastric slow waves originate from the ICCs. 2,6 The depolarization upstroke of the slow wave reduces the electrical threshold for circular smooth muscle contraction, and, in the appropriate situation, the amplitude of the circular smooth muscle contraction increases with the onset of the plateau potentials and action potentials. 7,8 The aborad propagation of slow waves linked to plateau potentials, with or without action potentials, is the electrophysiologic basis of gastric peristaltic waves, Fig 50.2. Thus, the slow waves linked with plateau or action potentials propagate through the corpus and antrum and create moving ring contractions that resolve in the antrum or at the pylorus in a terminal antral contraction. The pylorus provides an electrical barrier between the 3 cpm slow wave of the distal antrum and the 12 to 13 cpm slow wave of the duodenum. Intracellular electrical recordings from gastric smooth muscle cells. Intracellular recordings from smooth muscle cells from the different regions of the stomach, fundus to the mid-corpus to the terminal antrum, illustrate the electrophysiologic characteristics that distinguish these regions. Fig 50.3.1 Key features are: 1. Regional differences in resting membrane potential, which range from -48 to -75 mV, MV. 2. Regional differences in threshold for contraction, which range from -52 to -40 MV, and 3. The occurrence of plateau potentials with or without spike potentials. 1 The fundic smooth muscle cells are unique because their resting membrane potential lies at or above the threshold for contraction, -50 MV, a situation that promotes sustained smooth muscle contraction and ongoing fundic tone. Inhibitory vagal input to the fundus increases during swallowing and results in decreasing muscle tone associated with receptive relaxation and the accommodation of swallowed foodstuffs. 9,10 Fundic muscle tone decreases in proportion to the intensity and duration of the inhibitory neural discharge. In contrast to the fundus, intracellular recordings from the corpus indicate a lower resting membrane potential of -60 MV. The rapid upstroke depolarization in these cells is followed by a plateau potential that slowly returns to the baseline resting electrical potential. The plateau potentials are associated with circular muscle contraction activity in the corpus and antrum. 1 The plateau potential may be accompanied by action potentials in the corpus and antrum. Extrinsic stimuli such as release of acetylcholine or stretch of the stomach wall increases the amplitude and duration of the plateau potential and the occurrence of action potentials, resulting in contractions of varying force, as seen in the muscle of the terminal antrum. Depending on the excitatory neural stimuli and the amplitude of plateau potentials and the number of action potentials, peristaltic contraction waves of the circular muscle layer vary from very low amplitude contractions to high amplitude lumen-occluding contractions. At the pylorus, the plateau potentials have long durations with superimposed action potentials that result in closure of the pyloric sphincter in conjunction with the terminal antral contraction. 1 The membrane potential and the force of smooth muscle contraction also distinguish the fundus, corpus, and antrum. Fig. 50.4. The resting membrane potential of the fundus is approximately -50 MV and produces the sustained contraction and the resting tone of the fundus. 1 This fundic tone ensures a sensitive response to excitatory or inhibitory stimuli for relaxation or further contraction of the fundus. Receptive relaxation during ingestion of food is accomplished by these electrophysiologic attributes of smooth muscle in the fundus. In contrast, the resting membrane potentials of the corpus and antrum are -60 to -70 MV, respectively. In the presence of plateau potentials or action potentials, the membrane potential reaches -45 MV or less and smooth muscle contraction occurs. If the plateau potentials have higher amplitude, then contractions of larger amplitude or force occur. When the plateau potential and action potentials are linked to the propagating slow waves in the antrum, Fig. 50.5, then the moving ring contractions of the gastric peristaltic waves are formed. In conjunction with terminal antral contractions, the pyloric sphincter contraction prevents emptying of gastric content into the duodenum and results in retention of solid foodstuffs in the stomach. Thus, the peristaltic waves associated with terminal antral and pyloric sphincter contraction produce little or no emptying of the gastric contents from the stomach into the duodenum. In contrast, if the pylorus remains open during the gastric peristaltic wave, then an aliquot of nutrient chyme is emptied into the duodenum. Interstitial cells of Cajal ICCs are the pacemaker cells for the smooth muscle apparatus of the GI tract. 6,11,12 ICCs originate from c-KIT-positive mesenchymal cell precursors. 13 ICCs in the stomach are located in submuscular, intramuscular, myenteric, and subserosal layers of the gastric wall. 14-16 Figure 50.6 shows the anatomic relationships between the ICCs in the myenteric plexus, myex, the intramuscular ICCs, IMIX, the enteric neurons, and the circular smooth muscle cells. Myex are located between the circular and longitudinal muscle layers of the stomach and are the ICCs responsible for the generation of the slow waves. These ICCs spontaneously generate slow waves that are conducted into adjacent smooth muscle cells and cause depolarization and contraction of the smooth muscle by activating voltage-dependent, dihydropyridine-sensitive, L-type, calcium channels. 16,17 Increased amplitude of the plateau potential correlates with increased amplitude of smooth muscle contraction. The slow waves propagate. Fig. 50.1 Gastric electrical activity recorded from electrodes, A to D, positioned on the serosa of the stomach from the fundus to the antrum. Slow waves originate in the pacemaker region located at the juncture of the fundus and the corpus on the greater curvature. Note that the fundus does not have slow wave activity, electrode A. Slow waves propagate circumferentially and migrate distally to the pylorus approximately every 20 seconds, or 3 cycles per minute, CPM, dotted lines with arrowheads. GMA at 3 CPM can be recorded with cutaneous electrodes. The summed GMA recorded from electrodes positioned on the abdominal surface in the epigastrium is termed as EG, and the normal rhythm is 3 CPM. Modified from Cook KL. Electrogastrography. In, Schuster M., Crowell M., Cook, KL, editors. Atlas of Gastrointestinal Motility. Hamilton, Ontario, B.C. Decker, 2002. pp 185-201. Corpus fundus pyloric sphincter serosal electrodes 3 cpm slow wave, pace setter potentials, cutaneous electrodes 3 cpm egg waves 60 SEC 1 MV 500 V antrum ABCD duodenum pylorus pacemaker region lower esophageal sphincter chapter 50 gastric neuromuscular function and neuromuscular disorders 737 50 circumferentially and distally through the ICC network via gap junctions and entrain more distal ICCs with slower intrinsic frequencies to the higher slow wave frequency, the normal 3 cpm pacemaker frequency. ICCs are also located within the layers of the circular smooth muscle, IMIX, where they integrate and coordinate the spread of the slow wave and the smooth muscle contraction initiated by the MIX. 8 slow waves are not regenerated in the smooth muscle cells because the ion channels needed to generate and propagate slow waves are not expressed by gastric smooth muscle. In the corpus and antrum the MIX and IMIX form a continuous lattice-like network of interconnections that extend from the pacemaker region circumferentially and ally to the pylorus. The MIX establish the dominant pacemaker frequency, and IMIX carry the slow wave into the circular smooth muscle bundles to coordinate circumferential and aboral propagation of the contraction wave. The ICCs have innate rhythmicity that is based on their unique metabolism and fluxes in intracellular and extracellular calcium ions. 17,18 The most active area of depolarization and repolarization of the ICCs is in the pacemaker area of the stomach located between the fundus and the proximal corpus. The depolarization and repolarization of the ICCs is regenerated and propagated through the network of ICCs in a migrating wave front that moves from the pacemaker region on the greater curve through the corpus and antrum to the pylorus, proscribing the pathway of gastric peristaltic contractions. Excitatory input to the MICC, e.g., cholinergic stimuli, stretch, results in opening of calcium channels and depolarization of the smooth muscle cells with IMICC activation to coordinate the contractions of the circular muscle cells in time and space. Thus, the ICC networks provide the control of frequency and propagation velocity for the circular muscle contractions that comprise gastric peristalsis waves. The fundus of the stomach lacks slow waves. The IMIX and the fundus have a role in mechanoreception and act as sensory cells with interconnections to the vagal afferent neurons that innervate the fundus. 19 Fundic IMIX are also innervated by inhibitory vagal neurons that regulate tone in the fundus. 19 Thus, the ICCs also participate in the relaxation of fundic tone that occurs during accommodation. Normal human corpus and antrum have more than 5 ICCs/high power field, HPF. 20,21 Depletion of ICCs in the corpus antrum and loss of CD206 macrophages are associated with gastroparesis in patients with diabetes mellitus and idiopathic gastroparesis, IGP. 20-22 Severe depletion of ICCs is also associated with a variety of gastric dysrhythmias ranging from bradygastrias to tachygastrias and conduction defects. 21,23 Patients with DGP and loss of ICCs have more gastric electrical dysrhythmias, tachygastria, more upper GI symptoms, and poorer response to gastric electrical stimulation, GES, compared with patients with normal numbers of ICCs. 23 Interruption of ICC pathways from non-diabetic mechanisms also results in gastric dysrhythmias and ectopic pacemakers that are similar to gastric dysrhythmias found in patients with diabetes. 24 The loss of ICCs and DGP in mice is related to inflammatory infiltrates of M1 macrophages and increased production of inflammatory mediators such as interleukin-6. Whereas M2 macrophages appear to protect ICCs. 25,26 Corpus fundus 3 cpm slow waves plus plateau or action potentials, slow wave action potentials, plateau potentials, pace setter potentials, cutaneous 3 cpm egg waves 60 SEC 1 MV 500 V antrum moving peristaltic contraction ABCD duodenum pacemaker region pyloric sphincter lower esophageal sphincter Fig 50.2 gastric slow waves linked with plateau potentials, or action potentials, the electrophysiologic basis of gastric peristaltic waves. The plateau and action potentials occur during circular muscle contractions. Peristaltic waves originate in the pacemaker area. The frequency, 3 cycles per minute cpm, and propagation velocity, 14 mm/sec, of the gastric peristaltic waves are controlled by the slow wave, which leads the contraction from the proximal corpus to the distal antrum, as shown at electrodes A through D. The solid black lines and arrows indicate the circumferential and distal propagation of the peristaltic wave, which forms a ring contraction, small arrow, indicating a moving peristaltic contraction. Peristaltic contractions occur 3 times per minute, the frequency of the gastric slow wave. The increased GMA of the plateau potentials and action potentials linked with the slow wave results in increased amplitude of the 3 cpm waves recorded in the egg signal, thick black lines. The fundus does not participate in the gastric peristaltic contractions. Modified from Cook K.L. Electrogastrography. In, Schuster M., Crowell M., Cook, K.L., editors. Atlas of Gastrointestinal Motility. Hamilton, Ontario B.C. Decker, 2002. pp 185-201. 738 Part 6 Stomach and duodenum nervous system innervation. As reviewed earlier in Chapter 4, neurons of the ends populate the stomach wall from the fundus to the pylorus. 27 These neurons are located in the myenteric plexuses between the circular muscle and the longitudinal muscle layers. Neurons of the ends are also located in submucosal and subserosal plexuses. The ends provides local reflex circuits within the gastric wall as follows. 1. Sensory afferent neurons located in the mucosa are linked to 2. Interneurons in the myenteric plexus that are linked to 3. Efferent neurons that innervate the smooth muscle and glands to perform the gastric secretomuscular functions. 27 Release of excitatory neurotransmitters such as acetylcholine and substance P stimulates smooth muscle contractions, whereas inhibitory neurotransmitters such as nitric oxide and vasoactive intestinal polypeptide inhibit contractions. These enteric neural circuits within the gastric wall are programmed to modulate peristaltic contractions, in conjunction with ICC activity described earlier, by sequential inhibition of the distal smooth muscle segment and contraction of the immediate proximal segment of the stomach wall. 28,29 Serotonin in the bowel wall has a primary role in initiating and controlling peristaltic events. 27,28 Neurons of the ends are located in proximity to the MYICCs and IMICs. 30 The ends neurons provide additional control and modulation of contraction and relaxation of the gastric smooth muscle via cholinergic excitation and nitroergic inhibitory neurotransmission. Neurons of the ends form gap junctions with MYX and IMIX and provide crucial neural control that integrates slow wave activity and smooth muscle activity. Thus, postganglionic excitatory and inhibitory neurons innervate MYX to modulate gastric neuromuscular contraction and relaxation and provide chronotropic effects on the slow waves. Ultrastructural abnormalities in gastric neural cell bodies and nerve endings occur in patients with IGP and DGP. 31 The PNs and SNS modulate gastric neuromuscular activity. The vagus nerve provides the PNs input for the stomach, although approximately 80% of vagal fibers are afferent neurons. The afferent neurons respond to moment-to-moment contraction and relaxation, tone, of the stomach wall. 32 Efferent activity of the vagus nerve increases the release of acetylcholine, which most orad corpus mid orad corpus caudad corpus orad distal antrum pylorus B C D E F G H I 10 SEC 18 48 21 51 26 56 30 60 23 63 49 69 27 70 28 71 0 75 Fundus A Fig 50.3 Intracellular electrical recordings from smooth muscle from the fundus to the pylorus, A through I. Resting membrane potential in millivolts, MV, is shown on the vertical axis, and time is shown on the horizontal axis. Distinctive electrical characteristics in each region are shown. A. Spontaneous electrical activity in the fundic smooth muscle is absent. B through E. The resting membrane potential is less negative in the smooth muscle in the corpus compared with the antrum, F through H. Spontaneous upstroke depolarization is also recorded in the corpus and antrum, as well as in the pylorus, I. The upstroke depolarization in the smooth muscle is initiated by the interstitial cells of Cajal, see text. The upstroke depolarization is followed by the plateau potential and repolarization, D through I. The upstroke depolarization and plateau potentials are associated with contraction of the smooth muscle. Action potentials are superimposed on the plateau potentials in the terminal antrum and pylorus, G through I, and are associated with increased amplitude of smooth muscle contraction. From Sierzahusky J.H. Electrophysiological basis of gastrointestinal motility. In, Johnson L.R., editor. Physiology of the gastrointestinal tract. 2nd ed. New York, Raven Press, 1986. p. 383. Force of contractions, GF, 1.51.00.50.075 70 60 40 resting potential fundus membrane potential, MV, 30 corpus antrum Fig 50.4 relationship between membrane potential in millivolts, MV, and force of contraction in grams, GF, in gastric smooth muscle from the fundus, corpus, and antrum. The resting membrane potential in fundic smooth muscle is approximately -50 MV, a potential that generates muscle contraction and resting tone in the fundus. The resting membrane potential in the antrum smooth muscle is -70 MV, almost 30 MV below the threshold for smooth muscle contraction. When the resting membrane potential reaches -40 or -35 MV, the steep slope of the voltage contraction curve is observed in the corpus and antrum. From Sierzahusky J.H. Electrophysiological basis of gastrointestinal motility. In, Johnson L.R., editor. Physiology of the gastrointestinal tract. 2nd ed. New York, Raven Press, 1986. p. 383. Chapter 50 Gastric Neuromuscular Function and Neuromuscular Disorders 739-50 increases the amplitude of gastric contractions and stimulates secretion of gastric acid and pepsin. The SNS innervates gastric smooth muscle with neurons that travel with the splanchnic vasculature. SNS activity generally elicits inhibitory action on the smooth muscle via effects on the myenteric neurons of the ends. 33 The release of various hormones, ranging from CCK to gastrin, affects the neuromuscular activity of the stomach. Gut hormones produce their effect on smooth muscle, ICCs, and ends, as well as vagal efferent or afferent functions. These effects will be characterized under fasted and fed conditions and discussed in more detail later. Gastric neuromuscular activity during fasting. In the fasting state, electrical and contractile events of the corpus or antrum occur in a highly regular pattern termed the migrating myoelectrical, or motor, complex, or MMC. 34 The three phases of the MMC, as described by changes in intraluminal contractions, recur approximately every 90 to 120 minutes. Phase 1 is a period of quiescence wherein little or no contractile activity is recorded. In Phase 2 random, irregular contractions occur. Phase 3 of the MMC is a burst of regular, high-amplitude phasic contractions that last from 5 to 10 minutes, Fig 50.7a. Phase 3 contractions are also termed the activity front. The activity front migrates from the antrum to the ileum, a journey of 90 to 120 minutes duration. The three phases of the MMC occur regularly in the small intestine, although approximately 50% of the Phase 3 activity fronts originate in the stomach and then migrate through the small intestine. 35 The MMCs that originate either in the stomach or duodenum travel through the small intestine and terminate in the distal ileum. If fasting continues, then another Phase 3 activity front reappears in the antrum or duodenum at the 90 to 120 minute interval. The high amplitude, 3 per minute contractions of Phase 3 that develop in the distal antrum empty non-digestible, fibrous foodstuffs that remain in the stomach. Cyclic contractile activity associated with the onset of Phase 3 also has been identified in the lower esophageal sphincter, the sphincter of Oddi, and the gallbladder. The Phase 3 contractions correlate with rapid eye movement sleep and are related to a larger system of biological clocks. 36-37 MMCs develop after vagotomy, indicating that non-vagal mechanisms initiate and sustain MMC neuromuscular activity. Motilin is released during the intense phase 3 contractions that occur in the proximal duodenum. Gastric neuromuscular activity after a meal. 3 basic gastric neuromuscular activities occur during and after ingestion of solid foods. 1. Receptive relaxation to accommodate the ingested food. 2. Trituration of the ingested solid food by recurrent corpus antral peristaltic waves to produce chyme. And 3. Antral peristalsis with antropyloroduodenal coordination to empty chyme in small aliquots into the duodenum in a controlled manner for optimal digestion and absorption of the nutrients. Response to ingestion of solid foods. The neuromuscular work of the stomach in mixing, milling, and emptying food depends upon the physical characteristics, volume, and the fat, protein, and carbohydrate content of the ingested food. For example, 240 minutes of neuromuscular work by the normal stomach is required to empty 90% of a 255 kcal low-fat, egg substitute sandwich. 38 In contrast, only 35 minutes of gastric neuromuscular work is required to empty 70% of a 20 kcal 500 ml soup broth meal that was consumed in 4 minutes. 39 Figure 50.8 illustrates gastric neuromuscular activity required to receive, mix, and empty a solid meal. The spectrum of gastric work extends from fundic relaxation to gastric peristalsis to antropyloroduodenal coordination, the work that is needed to produce chyme and empty it into the duodenum. Ingestion of food abolishes the fasted state as regular 3 per minute. Gastric peristalsis begins in the corpus and antrum to mix the food. In the fed state, a pattern of continuous small bowel contractions with short runs of peristalsis over distances of 2 to 4 cm optimize digestion and absorption of nutrients. See Fig 50.7b. Solid food delivered from the esophagus into the fundus is associated with receptive relaxation of the fundus, the work of fundic muscle relaxation. As the fundic smooth muscle relaxes, larger amounts of solid or liquid food are accommodated in the fundus and proximal corpus with little or no increase in intraluminal pressure. Liquids, in contrast, are immediately distributed throughout the antrum and corpus. Emptying of liquids is discussed in the next section. Relaxation of the fundus occurs before the work of trituration in the corpus antrum and is a vagal nerve-mediated event that requires nitric oxide. 40 Figure 50.9 shows an example of the changes in intragastric volume during relaxation of the fundus and proximal corpus in response to a caloric meal. 41 Relaxation of the fundus and the stimulation of mechanoreceptors, stretch, mediated through imix in the fundic wall, activate vagal afferent neurons and vagovagal reflexes. These reflexes involve the nucleus of the tractus solitarius and efferent neurons from the dorsal motor nucleus of the vagus. Vagal excitatory neurons are inhibited and the vagal inhibitory neurotransmitters nitric oxide and vasoactive intestinal peptide are released to accomplish receptive relaxation. Tension 0.5 grams upstroke MP plateau phase 40-70 MVAB Fig 50.5 Relationships between smooth muscle contraction, tension, and membrane potential, MP. In these intracellular recordings from antral smooth muscle, the upstroke potential is the rapid depolarization, upstroke, event, followed by the plateau phase. The plateau potentials are associated with contraction of the smooth muscle cell, as shown in panel A. Note that an increase in the amplitude of the plateau potentials, red line in panel B, is associated with greater contractility, tension. During read polarization to the resting membrane potential, RMP, the contraction resolves. Modified from Sanders KM, Ordog T, KOHSD, Ward SM Properties of electrical rhythmicity in the stomach. In, Cook KL, Stern RM, Editors. Handbook of Electrogastrography. New York, Oxford Press, 2004. pp 1336. 740 Part 6 Stomach and duodenum. Other factors influence the muscle tone of the fundus. Antral distension, duodenal acidification. 42 Intraluminal perfusion of the duodenum with lipid or protein, and colonic distension all decrease fundic tone through various reflexes. The gastric reflex is mediated through an arc initiated by capsaicin-sensitive afferent vagal nerves and is mediated by 5-hydroxytryptamine 3, 5-HT3, gastrin-releasing peptide, and CCKA receptors. 43 Solid foods labeled with technetium are accommodated initially in the fundus and proximal corpus, and by obtaining frequent scintigraphic images, the distribution of the labeled solid meal can be followed over 4 hours using scintigraphic methods. 38 Figure 50.10 shows that immediately after ingestion of this solid meal, the food is accommodated and the majority of the meal is retained in the fundus and proximal corpus. Subsequently, contractions of the fundus press portions of the food into the corpus and antrum for trituration. This early postprandial period of accommodation and trituration that occurs before gastric emptying of the nutrients is termed the lag phase. The lag phase may last from 45 to 60 minutes for solid foods, but the duration of the lag depends on the thoroughness of chewing the food, the time required to ingest the meal, and the components of the meal. For a 255 kcal egg substitute test meal that is ingested in a 10-minute period, the lag phase is 30 to 45 minutes. Once portions of the meal have been triturated into 1 to 2 particles suspended in gastric juice, a linear phase of gastric emptying of the chyme begins. Recurrent gastric peristaltic waves mix saliva, acid, and pepsin with the chewed food and then mill the food to produce chyme. The normal peristaltic waves occur every 20 seconds, generated by 3 cpm slow waves linked to plateau and action potentials. In healthy subjects, approximately 60% of the egg substitute meal has emptied in 2 hours, and more than 90% has emptied at 4 hours. Fig 50.11. 38 Slow wave propagation in ICC-MI network. Electrotonic conduction of slow waves via gap junctions. Depolarization and activation of L-type Ca2 channels in SMCs. Neural inputs to ICC-SM and PDGFR cells. Conditions responses of SMCs to slow waves. SMC varicosity. Enteric motor neuron. SMCs, ICC, and PDGFR cells are electrically coupled, forming the syncytium. Together these cells generate the behaviors traditionally termed myogenic. Gap junctions. ICC-IM PDGFR. Spontaneous activation of pacemaker current. Fig 50.6 Relationships among ICCs, platelet-derived growth factor receptor alpha positive, PDGFR alpha plus, cells, smooth muscle cells, SMC, in the circular muscle layer, and motor neurons of the enteric nervous system. ICCs in the region of the myenteric plexus, ICC-MI, are pacemaker cells and spontaneously generate slow-wave depolarizations. Slow waves conduct to adjacent SMCs via low-resistance junctions, gap junctions, as shown by the curved arrow. Depolarization of SMCs leads to activation of L-type calcium channels, Ca2+ entry, and contraction of the SMCs coordinated by ICC intramuscular networks, ICC-IM. Thus, slow waves organize the contractile pattern of gastric smooth muscles into a series of phasic and propagating contractions. SMCs do not possess the ionic mechanisms necessary to regenerate slow waves, so the amplitude of slow waves decreases as slow waves conduct from SMC to SMC in a muscle bundle. Active propagation of slow waves from the dominant, i.e., highest frequency, pacemaker along the greater curvature of the gastric corpus to the pyloric sphincter requires continuous coupled networks of ICC-MIS, ICC-IMS, and SMCs. ICC-IMS are ICCs that lie within the circular layer of smooth muscle bundles. The ICC-IMS appear to be important in mediating neurotransmission because they form very close synaptic connections, gap junctions, with the varicose terminals of enteric motor neurons, short arrows. Postjunctional neural responses can be conducted from IMIX to muscle bundles. Thus, stimulation of excitatory enteric neurons leads to depolarization of ICC-IMS and increases the contractile responses of SMCs to slow-wave depolarizations initiated by the ICC-MI. Stimulation of inhibitory enteric neurons causes hyperpolarization and stabilization of membrane potential and tends to inhibit contractile responses to slow-wave depolarization. PDGFR-alpha and cells are another class of ICCs with distribution similar to ICCs that appear to mediate purinergic neurotransmission and SMC responses. Thus, these cells, the SMC, ICC, and PDGFR-alpha and form a syncytium of SIPs that produces rhythmic, stationary, and propagative contractions, myogenic, Events of the stomach. Chapter 50 Gastric Neuromuscular Function and Neuromuscular Disorders 741-50 During the linear phase of gastric emptying, each peristaltic wave empties from 3 to 4 ml of chyme through the open pylorus and into the duodenum. 44 Movement of chyme into the duodenum is usually, but not always, pulsatile due to the systole-like effect of antral peristaltic waves. 44 The volume of chyme delivered into the duodenum by each peristaltic wave is modulated by the configuration of the peristaltic wave, e.g., depth of contraction, length of the peristaltic wave, pressure within the stomach, and resistance to flow provided by the pyloric sphincter and duodenal contractions. 45,46 The gastric peristaltic wave delivers a larger stroke volume when the pylorus and the duodenum are relaxed to receive the aliquot of chyme, but the overall rate of calories delivered each minute to the duodenum is consistent at approximately 3 to 4 kcal/min. 43 As time elapses after ingestion of the meal, the chewed/swallowed food is continually redistributed from the fundus to the antrum for trituration. Some gastric peristaltic waves end at various points in the antrum and others end with a terminal antral contraction associated with closure of the pylorus that prevents the emptying of larger food particles or indigestible solids. These terminal antral and pyloric contractions result in delayed emptying of the solid particles in the corpus and antrum. The terminal antrum, the 3 to 4 cm of antrum immediately proximal to the pylorus, is also where the slow waves have the greatest amplitude and velocity. 5. In this manner, solid food particles that require further trituration are retained and subjected further to the milling effects of the recurrent peristaltic waves. The intragastric pressure and intraluminal pH values recorded after a healthy subject ingested an egg substitute meal are shown in Figure 50.12. Approximately 3.5 hours after the solid meal was ingested, high-amplitude contractions, greater than 65 mmHg, occur just before the pH suddenly increases from 1 to 6 as the wireless motility/pH capsule is emptied from the acidic antrum into the more alkaline environment of the duodenum. After the digestible components of the meal are emptied, strong antral contractions, phase 3-like contractions, empty the capsule from the stomach into the duodenum. 47 Thus, fibrous and indigestible materials are emptied by high-amplitude antral contractions, whereas the digestible nutrients in the chyme are emptied earlier by the lower-amplitude peristaltic waves during the linear phase of emptying. 48 The pylorus modulates the rate of gastric emptying by several mechanisms. Increased pyloric tone and isolated pyloric pressure waves prevent gastric emptying and promote retention of food for further milling. Pyloric contractions associated with terminal antral contractions are common during the lag phase when trituration is occurring. Once the linear phase of emptying of solids begins, the numbers of isolated pyloric contraction waves diminish as chyme is available for emptying via the gastric peristaltic waves. Neuromuscular dysfunction of the pyloric sphincter is associated with gastroparesis, is more common than previously appreciated, and is reviewed later. Response to ingestion of liquids. The gastric neuromuscular activity required to mix and empty liquids from the stomach is distinctly different from the emptying of solid foods. 39,49,50 Figure 50.13 shows three-dimensional US images of the stomach in a healthy subject during the fasting state and 10 minutes after the subject ingested 500 ml of soup. 39 The intragastric volume was approximately 40 ml during fasting and increased to 350 ml 10 minutes after ingestion of the soup, indicating the A. Pressure. CmH2O phase 3b 100 0 100 0 100 0 100 0 100 0 100 0 Time, min, 9:59:55 10 o'clock 1 antral 35246 antral duo pressure. CmH2O 100 0 100 0 100 0 100 0 100 0 100 0 8 28 38 40 antral 1 antral duo 2 antral 2 duo 1 duo 3 antral duo eat. Fig 50.7 Intraduodenal motor activity in a healthy subject. A. Fasted state, phase 3 contractions in the antrum, antral, and duodenum, duo. Intraluminal contractions in the antrum, channels 1, 3, and 5, and the duodenum, channels 2, 4, and 6, are shown. A phase 3 activity front with 3 per minute antral peristaltic contractions lasting almost 6 minutes is noted in channels 1, 3, and 5. The phase 3 activity front propagates distally and migrates past the duodenal recording ports. The frequency of contractions in the duodenum is approximately 11 or 12 per minute, the same as the frequency of the duodenal slow wave. After completion of the phase 3 contractions, the quiescence of phase 1 and lack of contractions are seen in the antrum. B. Fed state, the subject ingested a standard liquid meal, ensure. Contractions of variable amplitude are seen in the antrum and a series of relatively low amplitude, irregular contractions are noted in the duodenum, all of which represent the fed state and are in marked contrast to phase 3 activity during the fasting state shown in panel A, modified from Cook KL. The stomach. Manometry. In, Schuster M., Crowell M., Cook KL, editors. Atlas of Gastrointestinal Motility. Hamilton, Ontario, B.C. Decker, 2002. pp 135-150. 742 Part 6 Stomach and duodenum. Remarkable relaxation of the smooth muscle of the antrum and corpus, in addition to the fundic relaxation, that was required to accommodate this liquid volume. In contrast, solid meals are initially accommodated and retained primarily in the fundus and proximal stomach. Once accommodated, nutrient liquids are emptied into the duodenum in a controlled but more rapid rate compared with solid foods, which require trituration. Non-caloric liquid meals empty without the lag phase in a curve described as mono-exponential emptying, Fig 50.14, 0.50-51. Caloric dense liquids, on the other hand, are retained for longer periods in the antrum and are emptied slower than non-caloric liquids. Liquids are emptied from the stomach by a combination of 1. Pressure gradients between the stomach and the duodenum that produce flow of liquid into the duodenum, 2. Antral peristaltic contractions that produce a pulsatile pattern of emptying of liquids from the antrum into the duodenum, and 3. Duodenogastric reflux events that modify gastric emptying rates. 43,44 From a GMA viewpoint, ingestion of water until the point of fullness induces a brief frequency dip followed by return of normal 3 cpm activity recorded non-invasively in the electrogastrogram, EG, Fig 50.15. 0.52 The rate of gastric emptying of liquids is influenced by the volume, nutrient content, viscosity, and osmolarity of the ingested liquid. 39,45,46,51 These factors affect the neuromuscular activity of the stomach, which ultimately produces the rate of emptying. These factors are discussed later. Regulation of gastric neuromuscular activity after a meal. Gastric emptying rates are regulated to achieve a consistent, regular presentation of calories in the form of chyme to the duodenum in order to optimize secretion of pancreatic enzymes and bile appropriate for digestion of the contents of the chyme. Various gastric emptying rates are achieved by variations in the neuromuscular armamentarium of the stomach, fundic relaxation and contraction, the characteristics of gastric peristaltic contractions, temporary suspension of 3 cpm slow waves and the onset of gastric dysrhythmias, the coordination of antropyloroduodenal contractions and duodenal contractions, pyloric sphincter contraction and relaxation, and duodenal contractions that promote duodenogastric reflux. The attributes of a specific meal stimulate the appropriate gastric neuromuscular responses that affect the rate of gastric emptying. Table 50.1 lists gastric neuromuscular factors, meal-related factors, and other factors that modulate the rate of gastric emptying. The rate of gastric emptying is decreased by the temporary occurrence of gastric dysrhythmias, modulation of the amplitude and the propagation distances of antral contractions, enhanced contractions of the pylorus, and reduced antropyloroduodenal coordination. Meal-related factors that affect gastric emptying include the digestible components of the solids and liquids, fat content, nutrient density, viscosity, acid content, volume, osmolality, and indigestible foodstuffs. For example, foods with high fat content empty slower than foods with high protein or carbohydrate content. TGS are mixed with gastric lipase during the initial intragastric phases of digestion, see Chapter 102, and are broken down to fatty acids and monoglycerides or diglycerides before emptying into the duodenum. 53 The duodenum is exquisitely sensitive to diet-derived fatty acids. Longer-chain fatty acids, C12, exposed to the mucosa of the duodenum result in release of CCK. CCK relaxes fundic tone, decreases antral contraction, and increases pyloric tone, all of which result in delay in gastric emptying. In contrast, short and medium-chain fatty acids, C12, do not have antral peristalsis emptying duodenal resistance pyloric resistance corpus/antrum antral filling /mixing fundic contraction emptying fundic relaxation antropyloroduodenal coordination pyloric sphincter lower esophageal sphincter Fig 50.8 The spectrum of gastric neuromuscular work after ingestion of a solid meal. To receive the ingested solid foods and accommodate the volume of food without increasing intragastric pressure, the fundic smooth muscle relaxes, receptive relaxation. The fundus then contracts to empty the ingested solid food into the corpus and antrum for trituration and emptying. Recurrent corpus antral peristaltic waves mill the solids into chyme, which is composed of 1 to 2 mm solid particles suspended in gastric juice. Antral peristaltic waves, indicated by the ring-like indentation in the antrum, empty 2 to 4 ml of the chyme through the pylorus and into the duodenal bulb at the slow wave frequency of 3 peristaltic contractions per minute. Antropyloroduodenal coordination indicates efficient emptying of chyme through the pylorus, which modulates flow of the chyme by varying sphincter resistance. Contractions in the duodenum also provide resistance to emptying. Modified from Cook KL. Physiological basis of electrogastrography. In Cook KL, Stern RM, Editors. Handbook of Electrogastrography. New York, Oxford Press, 2004. pp 37-67. Chapter 50 Gastric Neuromuscular Function and Neuromuscular Disorders 743-50 These neuromuscular effects on gastric emptying rates. 46,54 CCK released from the duodenum also activates CCKA receptors on vagal afferent neurons with synapses in the nucleus tractus solitarius. 55 Neurons from the nucleus tractus solitarius ascend to the periventricular nucleus of the hypothalamus that participate in mechanisms of satiation, and descending vagal efferent neurons from the dorsal motor nucleus of the vagus inhibit gastric emptying and maintain fundic relaxation. The sensitivity of the duodenal mucosa to fat and other nutrients led to the concept of duodenal tasting and duodenal break, sensorimotor events that modulate gastric emptying of nutrients. 56,57 Monosaccharides in the duodenum stimulate the release of incretins such as glucagon-like polypeptide 1, which promotes insulin secretion to match increasing postprandial blood glucose levels and decreases antral contractions. 58,59 In order to harmonize the relationships between glucose absorption, glycemia, and insulin secretion, the gastric emptying of carbohydrates is highly regulated. 60,61 Hassler and colleagues showed that hyperglycemia decreases antral contractions and increases gastric dysrhythmias, a physiologic gastric dysrhythmia that decreases the rate of gastric emptying. Fig 50.16. 62,63 Hyperglycemia increases fundic compliance and decreases sensations related to fundic distension. 64,65 Blood glucose levels greater than 220 mg/dl result in decreased antral contractions, decreased gastric emptying, and induced gastric dysrhythmias. 62 All of which are gastric neuromuscular activities that reduce gastric emptying and reduce further exposure of the duodenum to nutrients. Hypoglycemia episodes are also associated with delayed emptying in patients with insulin-dependent diabetes. 65 The interaction between nutrients in the lumen and the regulation of the rate of gastric emptying continues in the later postprandial period as digestion and absorption of nutrients occur throughout the small intestine. For example, if diet-derived fatty acids or carbohydrates reach the lumen of the ileum, the so-called intragastric volume, ml, 500, 450, 400, 350, 300, 250, 200, 150, 150, 0, 30, 20, 10, 0, 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, 110, 120 time after the meal, min. Fig 50.9 Gastric accommodation of the fundus and proximal stomach in a healthy volunteer after a test meal. The intragastric volume, measured with a barostat balloon, increases from approximately 200 ml to approximately 450 ml during the 20 minutes after the meal is ingested. As the meal is emptied, the volume within the stomach slowly decreases over the 2-hour postprandial period. Relaxation of the proximal stomach and accommodation of the meal volume reflect vagal-mediated receptive relaxation. From TAC J, P. Savoux H, Coley B. ETAL role of impaired gastric accommodation to a meal in functional dyspepsia. Gastroenterology 1998, 115:1346-52. 0 minutes 120 minutes fundus antrum 180 minutes 240 minutes 30 minutes 60 minutes Fig 50.10 Gastric emptying of an egg sandwich. 1 minute scintigraphic images of a radio-labeled 255 kcal substitute egg meal in the stomach at time 0, 30, 60, 120, 180, and 240 minutes after ingestion are shown. The yellow and pink areas indicate regions of the stomach with higher isotope counts and more food than the other regions. Note the persistence of portions of the meal in the fundus at 120 minutes after ingestion. The meal is slowly redistributed from the fundus to the antrum for trituration and emptying. Only a small amount of the meal remains in the stomach by 240 minutes, and most of the labeled eggs are in the small intestine. 0, 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, 110, 120, 0, 30, 60, 90, 120, 150, 180, 210, 240% gastric retention time, min. Fig 50.11 Solid phase gastric emptying curve for 123 subjects after ingestion of a 255 kcal substitute egg meal, the same meal shown in figure 48.10. Note that only approximately 15% of the eggs are emptied in the first 45 minutes, the lag phase of gastric emptying of this meal. At 90 minutes, approximately 50% of the meal has been emptied and 50% is retained. By 240 minutes, more than 91% of the meal has been emptied. Modified from Tugas G, Aker EY, Abel TL, ETL. Assessment of gastric emptying using a low-fat meal, establishment of international control values. AMJ Gastroenterol 2000, 95:1456-62. 744 Part 6 Stomach and duodenum ileal brake is activated and gastric emptying is delayed. Infusion of nutrients into the lumen of the ileum delays gastric emptying. 66 Regulation of stomach emptying also is achieved by vagus nerve and splanchnic nerve activity that modulates the neuromuscular activities of the stomach described earlier. Vagal afferent nerves monitor neuromuscular function in the stomach moment by moment, and interactions between afferent vagal nerve activity and the nucleus tractus solitarius and synapses with the efferent vagal nerve output from the dorsal motor nucleus produce an ongoing interaction of CNS excitatory and inhibitory effects on the stomach. Gastric emptying is delayed during stress. Corticotrophin-releasing factor plays a role in the mediation of stress and inhibits gastric emptying through central dopamine 1 and dopamine 2 and vasopressin pathways in the periventricular nucleus. 67 Other factors that affect the rate of gastric emptying not already mentioned include rectocolonic distension, nausea and vomiting of pregnancy, and vection-induced motion sickness. 68 Stimulation of various areas in the CNS affects gastric neuromuscular function. Illusory self-motion, vection, induces antral hypomotility, tachygastria, and decreased gastric emptying. 68,69 A series of studies using the experience of illusory self-motion, a unique CNS sensory stimulation, showed that the onset of nausea was associated with tachygastria and increased levels of plasma vasopressin. 69,70 Gender affects the gastric emptying rate of a standard meal. Gastric emptying is significantly slower in healthy women compared with men. 71 Gender differences in gastric emptying rates may be related to fluctuations in sex hormones, but phases of the menstrual cycle, variations in estradiol and progesterone concentrations, have not shown consistent relationships with emptying measurements. 72 The rate of gastric emptying increases as body mass index rises, a relationship that may be relevant to the onset and maintenance of obesity. Gastric sensory activities. Free nerve endings in the stomach act as polymodal sensory receptors that respond to light touch or pressure. Acid and antrum corpus fundus AB Fig 50.13 Three-dimensional US reconstructed images from the stomach before and after a healthy subject ingested a 500 ml soup meal. A. In the fasted state, the intragastric volume is approximately 38 ml. B. 10 minutes after ingestion of the meal, the stomach volume is 350 ml. Note that the antrum, corpus, and fundus are now distended, indicating the marked relaxation of the smooth muscle required to accommodate this volume of liquid. Modified from Gilja O., Detmer Pr., Zhang J.M., ETAL intragastric distribution and gastric emptying assessed by three-dimensional ultrasonography. Gastroenterology 1997, 11338-49. 250 225 205 185 165 145 125 105 85 65 45 25 10 9 8 7 6 5 4 3 2 1 0 Pressure, mmHg, pH time, HR, pH pressure 0:030:28 0:57 126 155 224 252 321 354 19 430 Fig 50.12 Gastric contractions and intraluminal gastric pH recordings during the emptying of a 255 kcal egg substitute meal, the same meal shown in Figure 50.10, recorded with an ambulatory capsule pH and motility device. pH is shown on the right vertical axis, and pressure in mmHg is shown on the left vertical axis. The pH increases to approximately 3 for the first 45 minutes as gastric acid is buffered by the meal. The pH then gradually decreases to 1, and remains near 1 at about 3 hours after ingestion of the meal. Stomach contractions are generally of low amplitude, less than 10 mm Hg after ingestion of the meal. At approximately 3 hours and 40 minutes after the meal, the recorded pH increases abruptly to 7 and then decreases and remains stable at around 6. Prior to the abrupt increase in pH, there is a series of clustered, high-amplitude antral contractions, pressure. These antral contractions empty the capsule from the antrum, pH 1, into the duodenum, where the pH is 6 or more. The contractions that occurred during the 3 hours and 50 minutes required to empty the meal document the neuromuscular work required to triturate and empty this meal in a healthy subject. Chapter 50 Gastric Neuromuscular Function and Neuromuscular Disorders 745 50 Other chemical stimuli. Afferent neurons within the stomach are termed intrinsic primary afferent neurons, or IPANs. 73 cell bodies of IPANs reside in the submucosal or the myenteric plexus areas of the stomach wall. IPANs may be activated by serotonin release from local enterochromaffin cells. 27,74 The afferent information in the IPANs is used in local reflexes and provides input to vagal and splanchnic afferent neurons for vagovagal and spinal reflexes, respectively, to subserve transmission of visceral sensory information to CNS centers. Vagal afferent neurons whose cell bodies reside in the nodose ganglia connect with the nucleus of the tractus solitarius, and second-order neurons connect with higher center of the hypothalamus, and some inputs reach the cortex, where they are consciously perceived as visceral sensations, stomach emptiness or fullness, or symptoms such as nausea or abdominal pain. Fig 50.17. From the SNS, splanchnic or spinal primary afferent neurons in the gastric wall mediate pain sensations. Cell bodies of these neurons lie in the dorsal horn of the spinal cord, with second-order neurons that ascend via the spinothalamic and spinoreticular tracts in the dorsal columns. Sensory neurons are thin, myelinated A delta or unmyelinated C fibers. Spinal afferents include a population of unmyelinated C fibers. Capsaicin-sensitive unmyelinated fibers contain neuropeptides such as calcitonin gene-related peptide, vasoactive intestinal peptide, somatostatin, substance P, and neurokinin A. These fibers are considered to be the primary route of transmission for various pain stimuli from the gut to the CNS. These nerve fibers may respond to inflammatory mediators that also awaken silent nociceptive fibers. 75 In addition to interacting with IPANs, vagal afferent axons have multiple connections with the enteric neurons and innervate the circular muscle fiber bundles via connections with ICCs. 74 Vagal afferent neurons are also sensitive to chemostimuli via mucosal neurons and mechanosensitive neurons and ICCs in the muscle layers. CCK receptors on vagal afferent neurons are primarily activated by physiologic, mechanical and chemical stimuli from the stomach during fasting and fed conditions. These vagal afferents mediate the sensory response to intraluminal acid and fat. Acid may have a direct action on the nerve endings themselves. 42 Nausea is a common sensation that is often attributable to stomach neuromuscular dysfunction. 76 During the illusion of self-motion, gastric dysrhythmias develop as healthy individuals report nausea. 77 Plasma vasopressin levels increase in the subjects who develop nausea but do not increase in those who experience no nausea. 78 This brain-gut, gut-brain interaction during illusory self-motion illustrates the temporal relationships between the onset of gastric dysrhythmias in the periphery and acute, severe nausea experience of the subject. On the other hand, distension of the antrum, but not the fundus, with the balloon induces nausea sensations and gastric dysrhythmias in healthy individuals. 79 These studies show that gastric dysrhythmias originate in the antrum in humans and that stretch of the antral wall is another mechanism that elicits gastric dysrhythmias and nausea sensations from the stomach. Distension of the gastric antrum and corpus by the water load test, rather than a balloon, also elicits the gastric dysrhythmias and nausea in susceptible individuals. 52 The stomach and the regulation of food intake, hunger and satiety. Hunger is a basic human drive, a stressful condition that is eliminated or reduced by the ingestion of food. Hunger is also described as an uncomfortable emptiness of the stomach. The ingestion of food elicits relaxation of the stomach musculature, receptive relaxation, and accommodation of the physical volume of the meal. As these gastric neuromuscular events occur, hunger disappears and the comfortable, postprandial sensations of stomach fullness are experienced. The volume of food ingested suppresses hunger and stimulates the sense of fullness more than the calorie content of the meal. 80,81 Infusion of nutrients into the stomach induces a greater intensity of fullness or satiety compared with infusion of the same nutrients into the duodenum. The suppression of hunger is greater when nutrients are taken by mouth, indicating that CNS, oropharyngeal, and gastric neuromuscular factors are integrated to produce the comforts of normal postprandial stomach fullness. 82 Healthy individuals usually eat until they are reasonably full. The physiologic attributes of postprandial fullness are not 0.25 50 75 100 0.60 120 180 240 Gastric retention percent time, min. Solid phase meal 0.25 b 50 75 100 0.60 120 180 240 Gastric retention percent time, min. Liquid phase meal Fig 50.14 Gastric emptying of a mixed liquid and solid meal in healthy subjects who ingested 300 ml of radiolabeled water with 2 radiolabeled eggs and toast. A. Emptying rate for the solid phase of the meal. A short lag phase is noted before the linear phase of emptying, and by 60 minutes approximately 55% of the meal is emptied, 45% is retained. The lag phase may be shortened if the subject has taken a relatively long time to eat the meal or the solids require little trituration. B. Emptying rate for the liquid phase of the meal. Approximately 80% of the water is emptied, 20% is retained, at 60 minutes, as the liquid is rapidly distributed throughout the antrum and corpus. This is considered a mono-exponential liquid emptying curve. Modified from Maurer A., Parkman H.P., Knight L.C., Fisher R.s. Scintigraphy. In, Schuster M., Crowell M., Cook, K.L., Editors. Atlas of Gastrointestinal Motility. Hamilton, Ontario, B.C. Decker, 2002. pp 171-84. 746 Part 6 Stomach and duodenum completely known, but the physical stretch on the stomach walls, and changes in intragastric pressure, induced by ingestion of food and the secretion of gastric juice are in part responsible. 80,81 Subjects experience a dramatic change from the sensation of stomach emptiness at baseline to the sensation of stomach fullness after ingesting water over a 5-minute period. The average volume of water ingested to achieve fullness is 600 ml. In contrast, patients with functional dyspepsia, FD, ingest, on average, only 350 ml to feel full, indicating a disturbance in stomach wall relaxation and/or wall tension. 52 Similarly, fullness and satiety can be achieved by ingesting a nutrient drink until achieving maximum tolerated satiety. 83 The presence of acid or nutrients in the duodenum or an elevated blood glucose level decreases the stomach wall tension. 84,85 The ingestion of a solid meal initially elicits fundic relaxation, and little emptying of the food occurs during the lag phase. 100%, 90%, 80%, 70%, 60%, 50%, 40%, 30%, 20%, 10%, 0% time, min, time, min, time, min, time, min. Percentage distribution of egg power. Egg rhythm strips power running spectral analysis 0, BL. 10, 20, 30, 100%, 90%, 80%, 70%, 60%, 50%, 40%, 30%, 20%, 10%, 0%, 0, BL, 10, 20, 30, 100%, 90%, 80%, 70%, 60%, 50%, 40%, 30%, 20%, 10%, 0%, 0, BL, 10, 20, 30, 100%, 90%, 80%, 70%, 60%, 50%, 40%, 30%, 20%, 10%, 0%. 1 to 2.5 cpm, bradygastria, 2.5 to 3.75 cpm, normal, 3.75 to 10.0 cpm, tachygastria, 10.0 to 15.0 cpm. Duodenal response, 0, BL. 10, 20, 30 time, 1 minute between lines, frequency, CPM, 0639 12 15 B1A1B RESP. RESP. 500 VA water load 60 SEC 60 SEC Fig 50.15 Running spectral analysis of the egg signal and egg rhythm strips before and after ingestion of a water load in a healthy subject. The x-axis shows the frequencies in the egg signal in cycles per minute, CPM. The y-axis indicates time, and the peaks, or z-axis, indicate the power of the frequencies contained in the egg signal. The baseline egg rhythm strip, A, shows 3 CPM activity. The regularity and the amplitude of the 3 CPM egg signal is increased, B, after the subject ingested 750 ml of water load arrow, over a 5-minute period. The running spectral analysis shows relatively low power 3 CPM peaks at baseline, A1. After ingestion of the water load, the peaks initially disappear, the frequency dip, and then 3 CPM peaks emerge and are prominent until the end of the 30-minute recording, B1. This is a normal gastric myoelectrical response to the filling of the stomach with water and the subsequent emptying of the water. The 4 graphs in the insert show the percentage distribution of egg power in the 4 relevant frequency ranges during baseline, BL, and the 10, 20, and 30 minutes after ingestion of the water by the subject, green lines. Normal ranges are shown by blue lines. Note the initial decrease in the percentage of normal egg activity, 2.5 to 3.75 cpm, 10 minutes after ingestion of the water, the frequency dip, followed by increased percentages in the 3 cpm normal range 20 and 30 minutes after ingestion of the water. RESP, respiratory activity. Modified from Cook KL. Physiological basis of electrogastrography. In Cook KL, Stern RM, editors. Handbook of Electrogastrography. New York, Oxford Press, 2004. pp 37-67. Chapter 50 Gastric Neuromuscular Function and Neuromuscular Disorders 747 50 Sensations of fullness continue during the lag phase when the food is being triturated. Once the linear phase of gastric emptying begins, there is a progressive perception of decreasing stomach fullness and increasing stomach emptiness over time. 4 or 5 hours after a solid meal, the stomach is indeed empty and the healthy individual feels hungry once again. The physiologic mechanisms of hunger and satiety, and stomach emptiness and fullness, are under intense investigation. In the fasting state, plasma motilin levels increase during the phase 3 of the MMC, but correlations between the sensation of hunger and increases of motilin or onset of phase 3 have not been described. As discussed in chapters 4 and 7, ghrelin is a 28 amino acid peptide secreted from endocrine cells of the oxyntic glands in the gastric fundus. 86 Ghrelin levels also increase in the plasma during fasting, hunger, and stimulate food intake, probably acting via vagal afferent nerves. 87 Orexins or appetite-stimulating peptides are synthesized by neurons in the lateral hypothalamus, promote food intake, and stimulate gastric contractility, in the rat, by actions on the dorsal motor nucleus of the vagus with projections to the gastric fundus and corpus. 88 After ingestion of food, ghrelin levels decrease 89 and are profoundly suppressed after gastric bypass surgery. 90 Ghrelin also has promotility effects on the stomach and is being evaluated for the treatment of gastroparesis. 91, 92 Other hormones are candidates for important roles in the sensation of fullness or satiety, and these hormones are released after the ingestion of meals. CCK is released from the duodenal mucosa exposed to fatty acids. CCK receptors participate in fullness and nausea sensations elicited by intraduodenal lipid and gastric distension. 93, 94 Leptin is synthesized in the stomach and released after food ingestion. Circulating leptin reduces food intake via CNS regulation of the arcuate nucleus. 95 Glucagon-like polypeptide 1 enhances fullness after a standard meal, reduces antral motility, and increases gastric volume. 59, 96 Apolipoprotein A4 is released from the small intestine during absorption of TGS and decreases food intake and gastric motility, in part, via CCK and vagal afferent pathways. 97 Polypeptide YY is released from the ileocolonic area after meals and is an important mediator of the ileal brake effect. 98 And appetite suppression. 99, 100 The brain and these gut hormones are clearly linked in the regulation of food intake and the regulation of gastric neuromuscular activity that produces stomach emptying. 59, 100 The cephalic phase of gastric physiology is well known but has not been re-explored for many years. The sight, smell, and taste of food stimulate central vagal efferent activity that increases gastric acid secretion, gastric contractility, and increases 3-CPM GMA. 100-102 Sham feeding, during which the subject chews and spits out the test meal rather than swallowing it, elicits the cephalic vagal reflex. Sham feeding a warm hot dog on a bun elicits enhanced 3-CPM activity on the egg, whereas sham feeding a cold tofu dog, a food that the subjects considered disgusting, resulted in blunted or no increase in the 3-CPM myoelectrical activity, Fig. 50.18.103 Thus, sensory and emotional attributes of food during the cephalic phase of ingestive behavior also affect the neuromuscular activity of the stomach. Developmental aspects of gastric neuromuscular function. Gastric peristalsis appears between 14 and 23 weeks of gestation. Grouped or clustered peristaltic waves are evident by 24 Table 50.1 Factors that modulate the gastric emptying rate. Factors effect on gastric emptying rate. Gastric neuromuscular tachygastria delay, decreased fundic accommodation, acceleration, increased fundic accommodation delay, antral hypomotility delay, pylorospasm delay, intraduodenal discoordination delay, meal-related volume proportional to meal volume, increased acidity delay, increased osmolarity delay, nutrient density, fat, protein carbohydrate delay, tryptophan delay, undigestible fibers delay, small intestinal fatty acids and duodenum delay, duodenal tasting, duodenal break, fatty acids in ileum delay, ileal break, colonic constipation, IBS delay, other hyperglycemia delay, hypoglycemia, acceleration, illusory self-motion, vection, delay, proximal middle distal 0.02.55.07.510.0 time, min, proximal middle distal 0.02.55.07.510.0 time, min, AB Fig. 50.16 Electrical recordings from electrodes secured to the mucosa of the proximal, middle, and distal antrum in a healthy subject. A. 3 cycle per minute, CPM, electrical slow waves in the proximal, middle, and distal electrode leads. The slow waves are propagated in an aborad direction as indicated by the dotted lines. B. Disruption of propagation and the onset of a 5 to 6 cpm tachygastria in the distal lead during hyperglycemia, glucose clamping, with a blood glucose level of 240 mg/dl. Modified from Kolsky R. Hassler WL coupling and propagation of normal dysrhythmic gastric slow waves during acute hyperglycemia in healthy humans. Neurogastroenterol Model 2009, 21:492-99. 748 Part 6 Stomach and duodenum weeks. 104 The neuroregulatory mechanisms responsible for the coordination of antropyloroduodenal motility in gastric emptying are well developed by 30 weeks of gestation. 105 Egg recordings show normal 3 cpm activity in preterm infants delivered at 35 weeks that are similar to egg signals recorded in full-term infants. 106,107 On the other hand, egg recordings from premature infants, 35 weeks gestation, showed considerable tachygastria. 106 GMA matures further over the first 6 to 24 months of life and achieves full adult values by the end of the first decade. 108 The development of ICCs has been studied intensely because of the interest in gastric electrical rhythmicity, smooth muscle contractions, and gastric dysrhythmias. Labels for the tyrosine kinase receptor, c-KIT, and the availability of knockout mice lacking c-KIT have led to increased understanding of the development of ICCs. 109 The ICCs demonstrate differential development, with c-KIT expression on ICCs in the myics developing before birth, whereas ICCs in the deep muscular plexus, IMICCs, develop after birth. 110 ENS and ICC networks are not fully developed and are poorly coupled at birth, but progressive maturity of gastric rhythmicity and contractility occur during perinatal development. 106,107 The ENS and ICCs in the deep muscular plexus are closely related, whereas ICCs in the myenteric plexus can develop normally in the absence of the ENS. 110 Loss of ICCs in the pylorus is associated with loss of the inhibitory neural activity that may contribute to the development of pyloric stenosis in infants. See Chapter 49. 111 Assessment of gastric neuromuscular function. Gastric emptying rate. Clinical tests currently approved by the FDA to assess gastric neuromuscular function are scintigraphy tests to measure the rate of gastric emptying, the capsule motility device to measure gastric emptying, and egg devices to measure GMA before and after provocative test meals. These tests provide objective assessments of different aspects of the neuromuscular activity of the stomach pacemaker region, celiac ganglia, splanchnic nerves, vagus nerve, somatic nerves, dorsal root ganglion, CNS perceptions, discomfort, nausea, pain, visceral perceptions, discomfort, nausea, pain, somatic perceptions, discomfort, pain, A delta fiber, C fiber to CNS, visceral and motor reflexes, vertebral ganglia, T5 T9 motor, N IML, spinal cord, skin, stomach, gastric dysrhythmias, bradygastria, tachygastria, 60 SEC, efferent N, afferent N, afferent N, afferent N, C fiber, A delta fiber, C fiber, A delta fiber, spinothalamic tract, dorsal column, Fig 50.17, afferent and efferent neural connections between the stomach and CNS. The vagus nerve contains afferent nerves with A delta and C pain fibers with cell bodies in the nodos ganglia and connections to the nucleus tractus solitarius, not shown. Low-threshold mechanoreceptors and chemoreceptors stimulate visceral sensations such as gastric emptiness or fullness and symptoms such as nausea and discomfort. These stimuli are mediated through vagal pathways and become conscious perceptions of visceral sensations if sensory inputs reach the cortex. The splanchnic nerves also contain afferent nerves with A delta and C fibers that synapse in the celiac ganglia with some cell bodies in the vertebral ganglia, T5 T9. Interneurons in the white rami in the dorsal horn of the spinal cord cross to the dorsal columns and spinothalamic tracts and ascend to sensory areas of the medulla oblongata. These splanchnic afferent fibers are thought to mediate high-threshold stimuli for visceral pain. In contrast to visceral sensations, Somatic nerves such as those from the skin carry sensory information via A delta and C fibers through the dorsal root ganglia and into the dorsal horn and then through dorsal columns and spinothalamic tracts to cortical areas of somatic representation. Changes in gastric electrical rhythm, excess amplitude contractions, or stretch on the gastric wall are peripheral mechanisms that elicit changes in afferent neural activity, via vagal and/or splanchnic nerves, that may reach consciousness to be perceived as visceral perceptions, symptoms, emanating from the stomach. IML, intermediolateral nucleus, N, nerve. Chapter 50 Gastric Neuromuscular Function and Neuromuscular Disorders 749-50 in Health and Disease. Results of gastric emptying and GMA tests provide objective diagnoses of gastroparesis and gastric dysrhythmias and provide rational basis for treatments. Scintigraphy test meals labeled with radioisotope are available to assess the rate of gastric emptying. The seminal solid-phase gastric emptying protocol was a multinational study that used a 255 kcal technetium 99M, 99MTC, labeled egg substitute with bread and jam as the standard meal. 38 scans were obtained for 1 minute immediately after ingestion of the meal and at 30 minutes, 60 minutes, 120 minutes, 180 minutes, and 240 minutes in 123 healthy individuals. Delayed gastric emptying was defined as greater than 60% retention of the meal at 120 minutes and greater than 9% retention at 240 minutes. The 4-hour emptying test was superior to the 2-hour test because almost 20% of patients with suspected gastroparesis had normal emptying at 2 hours but abnormal emptying at 4 hours. 112 Pitfalls in the scintigraphic method for solid-phase gastric emptying studies include both improper binding of the isotope with the test meal, which results in rapid or normal emptying, and continuance of medications that may stimulate, e.g., metoclopramide, or inhibit, e.g., narcotics, anticholinergic agents, gastric smooth muscle contractions. These medications should be stopped 5 to 7 days before all gastric neuromuscular tests, if possible. Radiation exposure for the subject occurs with the scintigraphic tests, and multiple tests in the same subject are not advisable. Liquid phase gastric emptying tests, GETs, can be performed with indium 111-diethylenetriamine pentacetic acid, 99 mTC labeled water, or other liquids. Patients with unexplained nausea symptoms may have altered emptying of liquid meals, even if solid phase emptying is normal. 113,114 Capsule technology. Gastric emptying time of test meals is obtained from a small capsule that measures intraluminal pH and contractions. The capsule is swallowed with a standard test meal. During the postprandial period, measurements of luminal pH and contractions are transmitted to a receiver worn by the subject. In healthy subjects, the capsule is emptied from the stomach into the duodenum approximately 5 hours after ingestion of the egg substitute meal. Emptying of the capsule correlated with 90% emptying of the technetium-labeled egg substitute solid meal. The test had very good sensitivity and specificity in detecting gastroparesis. 115 Breath tests. Breath tests indirectly reflect gastric emptying of solid and liquid test meals. The solid meals are labeled with 1,3-C and include 1,3-C octanoic acid, 1,3-C acetate, or 1,3-C spirulina platensis. The 1,3-C octanoic acid breath test has been performed in many experimental protocols and is used widely in Europe for research and clinical studies. 116 The 13-C labeled food is emptied from the stomach and absorbed in the small intestine. The labeled nutrients are metabolized in the liver to 1,3-CO2, excreted in the lungs, and detected in breath samples. Breath samples are collected 45, 90, 120, 150, and 180 minutes after the meal in the 1,3-C spirulina test. 1,3-C is a stable isotope with no radiation risks. The 1,3-C breath tests are generally comparable to scintigraphy. 117 Pitfalls include spurious results in patients with malabsorptive conditions, liver diseases, or lung diseases that may preclude normal oxidation and excretion of the 13-C labeled foods. US transabdominal US techniques are used to measure antral diameter and antropyloroduodenal function. 118 Three-dimensional US methods show the intragastric distribution of the test meal and regional variations in gastric volume. 39 The technique is ideal with a liquid meal. The clinical application is limited by the high level of expertise required by the US operators. CT and MRI. These two techniques have been used to measure gastric emptying and demonstrate intragastric distribution of test meals. CT-AB time in 64 SEC intervals. Meal sham feeding 1357 frequency, CPM, 9-11-13-15 time in 64 SEC intervals. Meal sham feeding 1357 frequency, CPM, 9-11-13-15 Fig 50.18 Gastric myoelectrical response to sham feeding with tasty food, A, and disgusting food, B. A. Running spectral analysis, RSA, of the egg signal recorded while a healthy subject chewed a warm hot dog and spit it out into a paper bag, sham feeding. The increase in amplitude of the peaks in the normal 3 cycle per minute, CPM, range during sham feeding is the normal response. Meal indicates the actual ingestion of a hot dog. B. RSA of the egg recorded while a healthy subject chewed a cold tofu dog and spit it out, sham feeding. The subject felt disgusted during the sham feeding effort. Note the lack in increase of 3 CPM peaks during sham feeding the tofu dog compared with A. On both days, the subject then ingested a warm hot dog on a bun at meal. Note the subsequent increase in peaks at 3 CPM. Modified from Stern RM, Crawford He, Stewart WR, ETAL sham feeding. Cephalic vagal influences on gastric myoelectric activity. Dig Dis Sci 1989, 34:521-7. 750 Part 6 Stomach and duodenum and MRI technologies offer unique anatomic and functional views of the stomach in the fasting and postprandial periods. 119 Sequential antral contractions can be visualized. Because of expense and availability, these techniques are not used in clinical practice. Gastric contractions. Antroduodenal manometry. Antroduodenal manometry is an invasive technique wherein a water-perfused multi-lumen catheter is placed either through the nose or the mouth and advanced to a position where the proximal catheter ports are in the distal antrum and the distal ports are in the duodenum. 36 Placement of the catheter requires endoscopic or fluoroscopic aid. The recordings typically last for several hours in order to record phases 1, 2, and 3 of the MMC and several more hours to record postprandial contractions after the subject ingests a test meal. Antroduodenal manometry testing is not only invasive but also time-intensive and requires extensive assistant or physician time for performance of the test and interpretation of the data. Intraluminal manometry catheters detect only lumen-occluding contractions. 120 Intraluminal pressure transducer devices fail to record almost 50% of contractions in the corpus and antrum because the majority of postprandial peristaltic waves are not lumen-occluding contractions. Manometry catheters positioned in the duodenum can detect patterns of neuropathic or myopathic dysfunction. Capsule technology. The wireless ingestible capsule described previously measures contractions of the stomach wall. After a standard test meal, irregular contractions occur at 1 to 3 per minute and are consistent with manometric recordings from the antrum. 121 Several minutes of sustained high amplitude, antral contractions occur prior to the emptying of the capsule into the duodenum and some patterns are consistent with phase 3-like contractions recorded by antroduodenal manometry. In patients with gastroparesis, capsule studies showed a decreased motility index during the 20 to 30 minutes before emptying of the capsule, but a normal motility index in the 10 minutes before the capsule was emptied into the duodenum. These studies suggest that normal terminal antral contractions may be maintained even in patients with gastroparesis, whereas antral contractility required for trituration of digestible foodstuffs is abnormal. 122 Gastric myoelectrical activity GMA is recorded non-invasively using electrodes positioned on the abdominal surface. 4,123 The signal and egg summates the ongoing GMA, the slow wave activity during fasting and the summation of slow wave activity linked to plateau and action potential. Activity during the postprandial period. 124 In response to a water load or a nutrient load, the amplitude of the egg signal increases in the normal 2.5 to 3.7 cpm range, as determined by visual and computer analysis, e.g., an increase in the percentage of egg power or in the postprandial power ratio in the normal frequency range. 52, 125, 126 pitfalls for recording and analyzing eggs include failure to identify artifact in the signal and harmonics in the computer analyses. 127 Changes in the egg frequency and amplitude are key measures. After ingestion of most solid or liquid meals, a so-called frequency dip occurs in the first 10 to 15 minutes after the meal. The frequency dip reflects changes resulting from marked gastric relaxation and accommodation of the test meal related to the volume or the temperature of the meal. 128, 129 Several minutes after ingestion of the meal, the frequency of the egg signal returns to the middle of the normal 2.5 to 3.7 cpm range. Gastric dysrhythmias are associated with symptoms of nausea in subjects with motion sickness 78 nausea and vomiting of pregnancy 130-131 FD 52,132 and gastroparesis. 133-135 Gastric dysrhythmias include 0.5 to 2.5 cpm signals termed bradygastrias and 3.7 to 10 cpm signals termed tachygastrias, Fig 50.19. Recordings that have combinations of tachygastria and bradygastrias are termed nonspecific or mixed gastric dysrhythmias. 124,136 GMA can also be recorded from serosal electrodes placed during surgery or with mucosal electrodes placed during endoscopy. 5,21,63,137 Multiple channel serosal recordings using up to 128 channels have shown 3 cpm myoelectrical signals, as well as a variety of gastric dysrhythmias with similarities to cardiac dysrhythmias. 138,139 The amplitude and velocity of the human slow wave are greatest in the terminal antrum, the 3 to 4 cm of antrum immediately proximal to the pylorus. 5 These same studies further confirm the normal human slow wave frequency ranges from 2.5 to 3.7 cpm. Slow frequencies are also present from 1 to 2 cpm, bradygastrias, and from 4 to 9 cpm, tachygastrias, in patients with nausea with or without gastroparesis. 21,139,140 ABCD 500 V60 SEC 500 V500 V1000 V60 SEC 60 SEC 60 SEC Tachygastria bradygastria, low amplitude, bradygastria, high amplitude, normal. Fig 50.19 Gastric dysrhythmias recorded with egg methods. A. Tachygastria, an abnormally rapid signal at 6 cycles per minute, cpm, shown by dots. B and C, bradygastria at low or high amplitude 1 cpm wave, respectively. The 1 per minute waves are indicated by the solid curved lines, and the smaller waves in the egg signal represent respiratory activity. D. Normal 3 cpm egg signal is identified by dots. Chapter 50 Gastric Neuromuscular Function and Neuromuscular Disorders 751 50 Gastric Relaxation, Accommodation, and Volume Barostat Tests. Due to the spherical shape of the fundus and the proximal stomach, standard manometric catheters are not useful in recording intraluminal pressures after meals. The barostat balloon was designed to measure changes in tone, or gastric relaxation, and volume in the more spherical areas of the proximal stomach. 41 Intraballoon pressure is maintained with infused air during the baseline or fasting period with the balloon slightly distended. As the fundus and proximal stomach relax in response to the test meal, more air is concomitantly infused into the balloon to maintain the established baseline intraballoon pressure. 141 The volume of air that is infused to maintain baseline pressure is an estimate of the increased gastric volume that occurs as the proximal stomach relaxes. Barostat studies demonstrate abnormalities in fundic relaxation in almost 30% of patients with FD.41. The failure of fundic relaxation correlates with early satiety. Failure of fundic relaxation has also been recorded in patients with gastroparesis. 41,142,143 Because the barostat method is invasive and uncomfortable for patients, these studies have been limited to the research laboratory. Scintigraphy and other tests. Excessive or poor fundic relaxation in response to liquids and solids can be demonstrated with scintigraphy, US, and MRI. Single photon emission CT is a method that outlines the gastric wall before and after ingestion of a meal to determine changes in volume of the stomach. This method requires 4 injection of 99 mg per technetate to outline the gastric wall. The accommodation response can be identified with single photon emission CT. 144 Non-nutrient liquid and nutrient drink satiety tests. Non-nutrient liquids, the water load satiety test, and nutrient drink tests are used to assess overall gastric volume or gastric capacity and visceral sensations such as nausea, stomach fullness, or satiety in response to ingestion of these liquids. 52,83, 145, 146 often in conjunction with measures of GMA, accommodation, or emptying. In the water load satiety test, water is consumed over a 5-minute period until the subject feels full. 52, 146 in the typical caloric drink test, subjects drink 150 ml of the liquid, e.g., Ensure, every 5 minutes until maximum tolerated satiety is achieved, an endpoint that requires almost 30 minutes and results in the consumption of 800 to 1000 ml of the nutrient drink in healthy subjects. 146 in many healthy subjects, nausea and gastric dysrhythmia are evoked by the satiety drink test. Subjects with FD or gastroparesis ingest much smaller volumes of water or nutrient drink and report fullness, indicating impaired relaxation and accommodation of the stomach. 52, 83 Pyloric sphincter tests. The pyloric sphincter is a key regulator of gastric emptying, much like the lower esophageal sphincter is a key regulator of esophageal emptying. The pylorus is difficult to study in regards to fasting and postprandial function in awake subjects. The EndoFlip or endoscopic functional imaging probe, EndoFlip, Medtronic, Minneapolis, MN, is positioned across the pylorus during endoscopy to measure pyloric sphincter pressure, diameter, and distensibility. 147 Almost 30% of patients with gastroparesis have decreased pyloric distensibility compared with patients with normal gastric emptying. 148 Intraduodenal manometry. Intraduodenal manometry has been performed for many years, but it is difficult to keep pressure sensors as the catheters within the pyloric sphincter zone in a consistent and reliable fashion. Histopathologic studies in gastric and pyloric neuromuscular disorders. Efforts to define the histopathologic basis of gastric neuromuscular disorders have provided basic knowledge for the evolving field of neurogastroenterology. Most of the full-thickness specimens from the gastric wall have been harvested during the placement of GES devices or the placement of jejunostomy feeding tubes in patients with severe gastroparesis. Endoscopic biopsy techniques have been explored that provide full-thickness specimens that contain elements of circular muscle, ENS neurons, and ICCs. 149, 150 Histologic abnormalities in smooth muscle, neurons of the ENS, number or location of ICCs, and distribution of key neural or muscular receptors represent the potential underlying mechanisms of disordered gastric smooth muscle relaxation, peristaltic contraction, and gastric slow-wave activity. Loss of gastric ICCs, MIX and IMIX, was reported in patients with diabetic and IGP. 20,2131 Interestingly, similar loss of ICCs was shown in diabetic mice, and the ICCs were restored with intense insulin therapy, suggesting that the ICCs are not entirely destroyed in diabetes but dedifferentiate into immature myoblasts during prolonged hyperglycemia. 151 Damage to ICCs, ENS, and smooth muscle can also be seen in inflammatory and neoplastic conditions. 152-155 Other studies in humans have shown fibrosis of smooth muscle layers but intact myenteric plexus and vagus nerve in patients with DGP, loss of ICCs but minimal smooth muscle fibrosis, and inflammatory T lymphocyte infiltration in myenteric neurons. 156,157 Full-thickness biopsies from patients with IGP and DGP showed marked depletion of ICCs, a range of injury to enteric nerve cell bodies and endings, and preserved smooth muscle. 20-2331 Interestingly, more severe ICC depletion and enteric nerve injury were seen in the specimens from IGP patients compared with diabetics. 2031 The gastric smooth muscles in these specimens were generally normal. Thus, these disorders may be considered cagulopathies or gastric enteric neuropathies or, more likely in most cases, combinations of both. Further studies showed an immune infiltrate of CD204 macrophages was associated with decreased antral ICCs in patients with IgP and DGP.22. The loss of ICCs appeared to be more severe in the antrum compared with the corpus. The increase in CD204 macrophages was also associated with hemoxygenase deficiency and loss of ICCs, whereas replacement of hemoxygenase improved gastric emptying in mice. 158. However, a trial of hemin infusions in patients with DGP did not improve symptoms or gastric emptying. 159. Restoration of M2 macrophages in animals with gastroparesis resulted in increased numbers of ICCs and improved gastric emptying. 160. And suggests novel therapeutic pathways to treat gastroparesis. Finally, histochemical studies showed the pylorus in patients with gastroparesis contained decreased numbers of ICCs and increased fibrosis compared with control pyloric tissue. 161. These studies, as well as physiologic studies of decreased distensibility of the pylorus in gastroparesis 147, 148, 162, suggest an important role of the pylorus in regulating the rate of gastric emptying and thus in the development of gastroparesis. Results from histochemical studies of full-thickness gastric and pyloric tissue also provide new directions for understanding the neuromuscular dysfunction of the stomach and stimulate ideas for novel therapeutic approaches. Neuromuscular disorders of the stomach. Gastric neuromuscular disorders encompass a continuum of electrical and contractile dysfunction. At the mild end of the spectrum are gastric dysrhythmias, which are subtle electrical disturbances associated with mild to severe nausea symptoms, Fig. 50.20, studies showed these patients have a modest depletion of ICCs in the range of 3 to 5 ICCs/HPF. 140 abnormalities in 752 Part 6 stomach and duodenum. Relaxation of the fundus are associated with early satiety. At the severe end of the spectrum, antral hypomotility and profound gastroparesis are associated with prolonged postprandial fullness, vomiting, bloating, weight loss, and malnutrition that may require enteral or parenteral nutritional support. These patients have severe depletion of ICCs, 0 to 2 ICCs/HPF. 20-23 Patients with gastroparesis may also have multiple neuromuscular abnormalities, gastric dysrhythmias, dilated antrum, poor fundic relaxation, and gastric hypersensitivity or hyposensitivity due to vagal or splanchnic nerve dysfunction. 163-164 Symptoms associated with gastric neuromuscular disorders are summarized in Table 50.2. The pyloric sphincter is a key pathophysiologic factor in over 20% of patients with gastroparesis. 165 In these patients, the GMA is 3 cpm and indicates normal numbers of ICCs in the corpus and antrum. Gastroparesis and normal or hypernormal 3 cpm GMA are found in patients with fixed obstruction at the pylorus. 134 and in neuromuscular dysfunction of the pylorus. 147, 148, 166, 167 Thus, neuromuscular disorders of the stomach reflect a continuum of loss of ICCs but include obstructive gastroparesis. In the normal stomach 5 or more ICCs/HPF are present, the normal GMA is 3 cpm, and test meals are emptied at the normal rate. 20-24, subjects with chronic unexplained nausea and normal gastric emptying have 3 to 4 ICCs/HPF and a variety of gastric dysrhythmias ranging from tachygastrias to bradygastrias and various conduction defects, e.g., reentrant rhythms, conduction blocks. 140 Subjects with nausea and vomiting and gastroparesis have more severe depletion of ICCs with 0 to 2/HPF. And a variety of gastric dysrhythmias from tachygastria to bradygastria. 20-24 Finally, there is a subset of patients who have gastroparesis but have normal 3 cpm GMA. 134, 166, 167 These discordant findings, normal or hypernormal 3 cpm GMA and gastroparesis, indicate normal numbers of ICCs in the corpus/antrum, a pattern associated with pyloric dysfunction. 134, 147, 148, 168 Thus, pyloric dysfunction causing IgP may be due to a. Fixed, mechanical diseases such as stenosis due to peptic ulcer disease 134 or cancer 166 or b. Neuromuscular dysfunction such as pylorospasm 167, poor pyloric distensibility 147,148 or poor antropyloroduodenal coordination 168. Thus, a continuum of ICC depletion appears to relate to gastric dysrhythmias and gastric emptying rates and associated symptoms. Fig 50.21. Gastroparesis. Gastroparesis means paralysis of the stomach, as defined by the delayed rate of emptying of a standard test meal from the stomach in the absence of mechanical obstruction. Approximately 90% of the patients with gastroparesis have either diabetic, post-surgical, or IGP, but fixed obstructive or functional obstructive gastroparesis is important to identify because they are reversible. Table 50.3 lists the differential diagnosis for gastroparesis. Pacemaker region weak 3 cpm rhythm gastric dysrhythmias bradygastria dilated gastric antrum antral hypomotility gastroparesis vagal afferent nerve impaired fundic relaxation abnormal fundic emptying pylorospasm tachygastria 60s hypotensive lower esophageal sphincter Fig 50.20 The spectrum of gastric neuromuscular disorders. Gastric neuromuscular disorders range from abnormal fundic relaxation and emptying to gastric dysrhythmias and antral hypomotility and gastroparesis. Pyloric sphincter dysfunction, duodenal dysfunction, enteroduodenal discoordination, and vagal hypersensitivity may all be present in some patients with gastric neuromuscular disorders. CPM, cycles per minute. See text for details. Modified from Cook KL, Stern RM. Functional disorders of the stomach. Semin gastrointest Dis 1996, 7:185-95. Table 50.2 Categories of gastric neuromuscular disorders and treatment approaches based on gastric electrical and emptying test results. Category 1, Category 2, Category 3, Category 4. Test results. Gastric dysrhythmia and gastroparesis normal 3 cpm gastric rhythm and gastroparesis gastric dysrhythmia and normal emptying normal 3 cpm gastric rhythm and normal emptying Diagnosis severe gastric myoelectrical contractile disorder pylorospasm or fixed obstruction at the pylorus or duodenum electrocontractile dissociation gastric myoelectrical disorder visceral hypersensitivity non-gastric causes treatment nausea and vomiting diet prokinetic therapy antinauseant therapy gastrostomy tube / jejunostomy tube TPN accustomulation endoscopic therapies gastric electrical stimulation gastric pacemaker Botox / balloon dilation surgery for fixed obstruction nausea and vomiting. Diet prokinetic therapy antinauseant therapy nausea and vomiting diet prokinetic therapy antinauseant therapy nausea and vomiting diet antinauseant therapy antidepressant therapy drugs for fundic / antral relaxation, further workup for non-gastric causes, such as atypical GERD, cholecystitis, IBS, or disorders of the central or autonomic nervous system, see Table 50.6. See Table 50.5. CPM, cycles per minute. From Cook KL, Hassler WL, editors. Nausea and vomiting, diagnosis and treatment. Switzerland, Springer International Publishing, 2017. Chapter 50 Gastric Neuromuscular Function and Neuromuscular Disorders 753-50 The epidemiology of gastroparesis is just beginning to be understood. Data from Olmsted County, Minnesota, indicate an age-adjusted prevalence of definite gastroparesis of 9.6 per 100,000 persons for men and 37.8 per 100,000 for women. The incidence was 2.4 per 100,000 for men and 9.8 for women. The definite gastroparesis group was established on the basis of standard gastric scintigraphy results. 169 in diabetics. The incidence of gastroparesis was 1% in type 2 and nearly 5% in type 1, as compared with 0.1% for non-diabetic subjects in the population of Olmsted County. 169 Gastroparesis evolves over time in patients with diabetes, usually appearing after a duration of 10 years or more. In a series from Olmsted County, Minnesota, 5.2% of patients with type 1 diabetes and 1.0% of patients with type 2 diabetes developed gastroparesis over a 10-year period, whereas only 0.2% of control subjects developed gastroparesis. 170 in a community-based population of patients with type 2 diabetes mellitus, 6% of whites and 7% of African Americans had moderate intensity of symptoms associated with gastroparesis. 171 in contrast, in referral centers, gastroparesis is present in almost 50% of patients with type 1 and 30% of patients with type 2 diabetes mellitus. 172,173 hospitalizations related to gastroparesis have increased markedly since 2000. Many of the admissions for gastroparesis were patients with DGP with problems of glycemic control or infections. 174 A portion of these admissions were also due to surgical implantation of gastric stimulators for diabetic or IGP. 175 Diabetic gastroparesis. Many patients with long-standing type 1 diabetes mellitus develop DGP. These patients often have diabetes for more than 10 years. Erratic and elevated glucose levels, peripheral neuropathy, nephropathy, and cardiovascular disease. 164,175-177 patients with DGP have increased 5-year mortality compared with those without gastroparesis. 172,178 in a minority of patients the gastroparesis is the initial complication of their diabetes. One important manifestation of gastric emptying dysfunction in patients with insulin-dependent diabetes is erratic glucose control, especially with unexpected hypoglycemic episodes in the postprandial period if the usual insulin doses are administered before meals. When postprandial insulin levels increase following insulin injection and yet gastric emptying is delayed, nutrient delivery into the duodenum and intestinal glucose absorption are delayed. Thus, plasma glucose levels decrease in response to the insulin treatment and symptomatic hypoglycemia develops unexpectedly. 177 Acute hyperglycemia, greater than 220 mg/dl, is associated with tachygastrias and delayed gastric emptying in healthy subjects and in patients with diabetes. 63,179,180 Hyperglycemia is also associated with loss of ICCs. 151 Antral hypomotility. 181 Isolated caloric contractions. 182 Gastric dysrhythmias. 63,179 and impaired prokinetic action of prokinetic drugs like erythromycin. 183 Relatively minor increases in the blood sugar level, even elevations within the physiologic range, delay gastric emptying in normal volunteers and diabetic patients. 180 Acute hyperglycemia produced by glucose clamp methodology elicits fullness, decreased antral contractility, gastric dysrhythmias, blunts the contractile caloric response to intraduodenal lipid infusion, and modifies upper GI sensations. 63,179,180 Gastric neuromuscular abnormalities documented in patients with type 1 diabetes include abnormal intragastric distribution of food. 184 Reduced receptive relaxation and accommodation. 185 Reduced incidence of the antral component of the MMC, antral dilation, postprandial antral hypomotility. 181 and electrical dysrhythmias. 133,173,186 in patients with DGP. The ICCs are depleted and enteric nerve endings are abnormal. 20,21 Pathologic findings that help explain the mechanism of gastric dysrhythmias and poor gastric contractile responses to test meals. Loss of the antral normal 3 cpm GMA and normal gastric emptying. Normal 3 cpm GMA and gastroparesis, obstruction, gastric dysrhythmias and normal gastric emptying. Gastric dysrhythmias and gastroparesis. Interstitial tsela of Kagel/HPF greater than 5,4,3,2,10 numbers of ICCs and caloric dysfunction. In contrast, in the obstructive gastroparesis subtype, normal 3 cpm GMA is present, and reflects normaling, whereas severe ICC depletion, 1 to 2 ICCs/HPF, is associated with gastric dysrhythmias and gastroparesis. Modest depletion of ICCs, 3 to 4 ICCs/HPF, is associated with gastric dysrhythmias and normal gastric emptyux, 5/high power field HPF, are associated with normal 3 cpm GMA and normal gastric emptying. Fig 50.21 Schematic of relationships among ICCs, GMA, gastric emptying is shown. Normal numbers of 754. Part 6. Stomach and duodenum. Phase 3 contractions results in poor emptying of fibrous debris in the stomach and is the neuromuscular basis for the formation of bezoars. See Chapter 28. Thus, the progressive neuromuscular dysfunction of the diabetic stomach reflects the effects of chronic hyperglycemia and superimposed, intermittent hyperglycemia episodes on the gastric ends and ICCs. Gastric smooth muscle dysfunction is another mechanism of delayed gastric emptying in some patients with diabetes. Gastric smooth muscle contractility in diabetic rats is reduced in response to electrical stimulation. 187 The inhibitory effect of hyperglycemia on stem cell factor has a role in the reduction in ICCs and smooth muscle contractility. 188 In humans with DGP, the smooth muscle is intact but has a thickened basal lamina. 20,21 Diabetic patients with gastroparesis were treated with insulin pump therapy with continuous glucose monitoring for 48 weeks. The hemoglobin A1C decreased significantly, by 1.1%. And the symptoms associated with gastroparesis recorded with the Gastroparesis Cardinal Symptoms Index decreased by 23% compared with baseline. 189 Thus, aggressive insulin treatment in these patients helped glucose control and gastroparesis symptoms. 189 Pylorospasm may cause a form of obstructive DGP. 167,190 In these cases the neuromuscular function of the corpus and antrum is intact, but functional obstruction at the pylorus due to pylorospasm or incomplete relaxation of the pylorus causes delayed emptying. These patients have normal or increased 3 cpm GMA, a discordant finding in patients with gastroparesis that suggests the possibility of gastric outlet obstruction from functional or mechanical causes at the pylorus. Fig 50.22a, see section on obstructive gastroparesis later. 134,190 Almost 25 million people in the USA have type 2 diabetes mellitus, and many have unsuspected or subclinical gastroparesis. 191 In tertiary centers, the incidence of gastroparesis in type 2 diabetes mellitus ranges from 30% to almost 50%. 191 Patients with type 2 diabetes differ significantly from patients with type 1 diabetes in that they have insulin resistance, the diabetes appears later in life, obesity is common, early satiety is more common, gastroparesis is less severe, and the hyperglycemia has been present longer before diagnosis compared with patients with type 1 diabetes. 191-194 In the early stages of type 2 diabetes mellitus, gastric emptying may be accelerated. 195 A study from the National Institutes of Health Gastroparesis Consortium showed patients with type 1 diabetes mellitus are younger, have more severe delays in gastric emptying, and have more hospitalizations compared with type 2 diabetes mellitus and gastroparesis. Type 2 diabetes mellitus patients with gastroparesis are older, have milder delays in gastric emptying, and more severe early satiety. 193,194 Experimental diabetes in non-obese diabetic mice and in the type 2-like diabetes DB/DB mouse leads to gastroparesis. Neuronal nitric oxide synthase, NNOS, is reduced in the diabetic mouse stomach and is associated with antropylorospasm and gastroparesis. 196 Notably, NNOS was decreased 40% in human stomach tissue from patients with DGP compared with control values. 20 Treatment with insulin or sildenafil restored NOS and was associated with reversal of the delay in gastric emptying in the diabetic mouse. Hypomotility of the fundus and hypercontractility of the pylorus were found in DB/DB mice. 197 In other studies of diabetic mice, the loss of ICCs was associated with electrical dysrhythmias, delayed gastric emptying, and reduced neurotransmission in gastric smooth muscle. 198 Post-surgical gastroparesis. Gastroparesis occurs in a subset of patients undergoing subtle or radical stomach operations that range from vagotomy to fundoplication to antrectomy. Truncal vagotomy produces complex effects on the neuromuscular function of the stomach. After vagotomy, the fundus fails to relax normally after meals, resulting in rapid filling of the antrum. 199 Vagotomy is also associated with gastric dysrhythmias. 3 Decreased antral contractions, and poor entropyloroduodenal coordination. 200 Truncal vagotomy performed during ulcer operations requires a pyloroplasty to reduce sphincteric resistance to antral outflow because antral contractions become weak and peristalsis is disrupted. Most patients recover from the effects of vagotomy. But in patients undergoing extensive resection of the antrum and corpus, prolonged symptoms and chronic gastric neuromuscular dysfunction are likely. Lower esophageal resection for esophageal cancer includes resection of the fundus. The vagus nerves are transected and the fundic reservoir is lost. Pyloroplasty is performed to facilitate gastric emptying, but the loss of the fundus and variable amounts of the corpus, that may encompass the pacemaker region, often leads to chronic nausea, gastric dysrhythmias, and gastroparesis. Antral resections with Bilroad I, Bilroad II, or Roux-en-Y gastrojejunostomy for gastric tumors or PUD may lead to profound neuromuscular dysfunction of the stomach. 201,202 Critical amounts of the corpus antrum, including unknown amounts of the gastric wall containing the pacemaker region, required for normal gastric neuromuscular activity may be resected, and the efficacy of trituration by the corpus and antrum may be markedly reduced. Ingested food is retained in the remnant fundus and fails to empty into the corpus. 203. The corpus fails to mix and empty gastric contents even though the anastomosis is widely patent. The Roux-en-Y gastroenterostomy operation may result in the Roux syndrome in which postprandial pain, bloating, and nausea develop. Delayed gastric emptying is due to functional obstruction by the Roux limb as the neuromuscular dyssynchrony within the Roux limb prevents emptying of the stomach. 204,205 After vagotomy and antrectomy, patients may develop the dumping syndrome described later. In the gastric sleeve resection for obesity, 2/3 of the stomach is removed including portions of the fundus, corpus, and antrum. The pylorus is retained. After the sleeve resection, gastric emptying of liquid and solid test meals is accelerated, but there are few studies available for review. 206,207 Fundoplication is commonly performed to treat gastroesophageal reflux disease that fails to respond to medical therapy, see Chapter 46. Post-fundoplication gastroparesis and early satiety, bloating, prolonged fullness, and nausea may occur. These patients have altered fundic relaxation, delayed gastric emptying, and gastric dysrhythmias, possibly on the basis of vagal nerve injury during or after the fundoplication procedure. 208,209 Because gastric emptying studies are infrequently performed before this operation, it is not known how many of the patients already had gastroparesis before the fundoplication. 209 Table 50.3 Causes of gastroparesis Diagnosis Incidence, %. IGP*, 20% have obstructive gastroparesis, 40 diabetic gastroparesis, type 1 and 2 diabetes mellitus, 20% have obstructive gastroparesis, 35 post-surgical gastroparesis, antrectomy, vagotomy, fundectomy, fundoplication, 20 ischemic gastroparesis. 1 Miscellaneous causes. 4 possibly post-viral, drug-induced, degenerative or inflammatory processes of enteric nerves, interstitial cells of Cajal, or smooth muscle. Chapter 50 Gastric neuromuscular function and neuromuscular disorders. 755 50 Ischemic gastroparesis. Chronic mesenteric ischemia is a rare but reversible cause of gastroparesis. 210 These patients present with the symptoms associated with gastroparesis. Ischemic gastroparesis is distinct from acute mesenteric ischemia, which presents as an abdominal catastrophe with an acute abdomen and gangrenous small intestine, see Chapter 118. Chronic mesenteric ischemia is usually due to progressive atherosclerosis or hyperplasia of the intima of the arteries of the celiac, superior mesenteric, or inferior mesenteric artery. Collaterals of these obstructed arteries form over time so that neuromuscular function of the stomach is preserved, at least for some time. Bypass graft surgery or dilatation of the stenotic arteries results in resolution of symptoms, eradication of gastric dysrhythmias, and reversal of gastroparesis. 210 Thus, ischemic gastroparesis is potentially a reversible form of gastroparesis and should be suspected in patients with gastroparesis, weight loss, and a history of peripheral vascular disease, cerebral vascular disease, or myocardial infarction. An abdominal bruit is present in approximately 50% of patients. There are other uncommon forms of mesenteric vascular compromise, e.g., the median arcuate ligament syndrome, that may result in decreased blood flow to the stomach. 211 Release of the arcuate ligament and restoration of blood flow have been associated with improvement in gastric emptying. On the other hand, superior mesenteric artery syndrome is not accepted as a cause of mechanical obstruction that leads to gastroparesis, nausea, and vomiting. Patients with idiopathic, discussed below, diabetic, or post-surgical gastroparesis may have a subtype of gastroparesis, obstructive gastroparesis, due to pyloric dysfunction. This subtype is important because endoscopic and surgical treatments are directed towards the pylorus. Fixed pyloric obstruction. Fixed obstructive gastroparesis refers to delayed emptying due to mechanical obstruction at the pylorus or duodenal bulbar or post-bulbar area by tumor, chronic peptic ulcer or inflammation, rings, or webs. 134,166 The obstructions may be distal to the duodenal bulb in the descending and transverse portions of the duodenum. These patients have gastroparesis and normal or hypernormal 3 cpm GMA. 134 Figure 50.22a shows an example of high-amplitude 3 cpm GMA waves recorded in an egg test in a patient with pyloric outlet obstruction due to chronic PUD and fixed narrowing of the pylorus. In these patients, the smooth muscle ends and ICCs of the corpus antrum are intact, but the normal recurrent gastric peristaltic waves meet sustained resistance at the point of obstruction, the pylorus, and the emptying of solid food is delayed. In contrast, most patients with IGP have bradygastria, tachygastria, or mixed gastric dysrhythmias indicating both electrical and contractile dysfunction, see Fig 50.22b. However, if patients with IGP or DGP have 3 cpm GMA, then causes for the gastroparesis should be considered. Gastroparesis may actually be obstructive gastroparesis due to pyloric stenosis or post-duodenal bulb cancer. 134,166 Surgical correction of the fixed pyloric obstruction with resection and Billroad I or Billroad II gastrojejunostomy is necessary to correct obstructive gastroparesis, although some patients may respond to balloon dilation of strictures, see Chapter 53. In patients who have had prolonged mechanical obstruction of the pylorus, the stomach may dilate, smooth muscle contractions weaken, but 3 cpm GMA is present, all which may represent a form of gastroparesis due to electrical contractile dissociation. Functional pyloric obstruction. A more subtle type of gastric outlet obstruction occurs in pylorospasm. The spasm of the pylorus may cause postprandial right upper quadrant abdominal pain in the setting of gastroparesis-like symptoms. Pylorospasm of the pylorus prevents normal gastric peristaltic waves from emptying chyme into the duodenum. Dyscalasia was described by Fisher et al. To indicate poor antropylora coordination when the pylorus closes prematurely and inhibits emptying. 168 Thus, the rate of gastric emptying is delayed and gastroparesis is diagnosed in the presence of pyloric dysfunction. However, these patients with gastroparesis have normal 3 cpm GMA as recorded in the egg because the neuromuscular apparatus of the corpus antrum is intact. Fig 50.23. AB frequency, CPM, frequency, CPM, 3691215 3691215 Power egg rhythm strips 1 MV 60 SEC time, 1 minute between lines. 01 MV Power egg rhythm strips 500 VA 60 SEC time, 1 minute between lines. A1 water load, 250 ml. BB1 Fig 50.22 RSA of the egg in patients with obstructive and idiopathic gastroparesis. A. RSA recording from a patient with gastroparesis due to mechanical obstruction at the pylorus secondary to chronic PUD. Note the persistent high amplitude 3 cycle per minute. CPM, waves in the egg rhythm strips and the uniform and unvarying peaks at 3 cpm in the RSA, findings that would not be expected in a patient with gastroparesis due to electrical and contractile dysfunction. B. RSA and egg rhythm strips. Recordings in a patient with IGP. Note that the egg rhythm strips show a 7 to 8 cpm tachygastria before, B, and after, A water load test. The RSA shows multiple peaks in the 7 to 8 cpm tachygastria range and few peaks in the normal 3 cpm range. This patient had electrical and contractile abnormalities of the stomach as documented by the tachygastria and gastroparesis. Modified from Brezina R.J., Cook K.L., Bingaman S. Gastric myoelectrical activity in patients with gastric outlet obstruction and idiopathic gastroparesis. AMJ Gastroenterol 1998, 93:1803-9. 756 Part 6 Stomach and duodenum. In these patients, dilatation of the pylorus with a 20 mm balloon for 2 minutes or botulinum toxin A injections into the pylorus decreased postprandial symptoms. 167 In patients with more than 3 successful endoscopic pyloric treatments, pyloroplasty was recommended and completed. All patients had improved symptoms and increased gastric emptying rates. 212 Finally, some patients with obstructive gastroparesis may have normal 3 cpm egg signals and poor gastric emptying on the basis of electromechanical dissociation. In these situations, it is possible that mix or imix that generate slow waves may be normal, but enteric neurons and/or smooth muscle dysfunction are present. Idiopathic gastroparesis. In over 1/3 of patients, the gastroparesis is idiopathic, IGP. 213 Patients with IGP tend to have more abdominal pain compared with DGP, but nausea and vomiting are the dominant symptoms driving visits to gastroenterologists. 214 In 15% of these patients, an acute febrile illness preceded the diagnosis of gastroparesis by many months. 215 These herald illnesses are frequently described as flu-like with fever and nausea and vomiting. Patients often date the onset of their nausea from that point. Norwalk virus, HSV, and EBV infections have been documented in patients with sudden onset gastroparesis and normal immune systems, whereas other patients are immunocompromised. 216 Post-viral gastroparesis may resolve completely over 1 to 2 years. New histologic studies from the National Institutes of Health Gastroparesis Consortium indicate that patients with IGP have more severe depletion of ICCs and loss of enteric nerve endings compared with DGP. 2031 Loss of ICCs in knockout mice is associated with gastric dysrhythmias. 217 Gastric dysrhythmias are common in patients with IGP, see Fig 50.22b. and indicate severe loss of ICCs in the corpus/antrum. 20-22 Other historical clues to the genesis of IGP are exposures to 1. Multiple courses of antibiotics, e.g., treatments for chronic ear or sinus infections, 2. Anesthetic agents for a variety of common operations, and 3. Food poisoning like illnesses of unknown cause. However, as discussed earlier, almost 20% of patients with IGP have normal 3 cpm GMA, the functional obstructive subtype of gastroparesis. IGP is diagnosed in patients who have dyspepsia-like symptoms, delayed gastric emptying, and gastric dysrhythmias, but no primary causes of gastroparesis such as diabetes, ischemia, or gastric operations are present. Many other diseases and disorders, including intestinal pseudoobstruction, discussed later, paraneoplastic diseases, autonomic nervous system abnormalities, thyroid and adrenal disease, or CNS diseases, may cause or 250 ml water frequency, CPM, time, min, 03691215 after water load baseline Fig 50.23 egg rhythm strips from baseline and 30 minutes after the water load test from a patient with IGP. The x-axis indicate frequencies in the egg signal, the y-axis indicates time, and the z-axis indicates the power of the frequencies represented in the peaks. Clear 3 cycles per minute, CPM, waves are seen in the rhythm strips, right, and reflect normal slow waves and numbers of ICCs. Computer analysis of the egg recording, left, shows regular 3 CPM peaks in the running spectral analysis. This is the obstructive subtype of gastroparesis with gastroparesis and normal 3 cpm GMA. Mechanical obstruction at the pylorus was excluded at endoscopy. Chapter 50 Gastric neuromuscular function and neuromuscular disorders 757-50 are associated with gastroparesis. If these disorders are identified, then the gastroparesis may be secondary to these specific diseases. Gastric neuromuscular dysfunction associated with other GI disorders. FD symptoms include epigastric discomfort or pain, early satiety, fullness, nausea, and vomiting in the setting of normal upper endoscopy and normal routine laboratory tests. FD is divided into an epigastric pain syndrome and postprandial distress syndrome, PDS, the latter comprising 80% of the FD patients. 218 One of the dominant symptoms that patients with FD have is recurrent and unexplained nausea and vomiting. This is a debilitating symptom with a large differential diagnosis, box 50.1, and these disease categories should be considered in the evaluation of the patient with nausea and vomiting. If the patient has one of these disorders, then FD is not the diagnosis. Importantly, postprandial distress symptoms are also similar to symptoms associated with gastroparesis. 219 The gastric pathophysiologic mechanisms that drive FD symptoms remains an area of intensive investigation, see Chapter 14. If endoscopy is normal, then FD symptoms may be caused by several neuromuscular disorders of the stomach. For example, gastroparesis is associated with FD symptoms. 81% of patients with IGP have symptoms similar to those defining PDS. 214 The diagnosis in these patients is IGP with associated symptoms. 17 to 40% of patients with FD have gastroparesis, and 40% to 62% have gastric dysrhythmias. 52, 132 Treatments can be designed to address the several pathophysiologic abnormalities of gastroparesis. If primary causes for gastroparesis such as functional or fixed pyloric outlet obstruction, diabetes, or gastric operations have been excluded, then these patients have IGP. Studies show patients with FD or gastroparesis-like symptoms have the same symptoms and same severity of symptoms as patients with gastroparesis. 219 Improvement in gastric dysrhythmia and FD symptoms was reported in patients treated with cisapride. 220-222 Thus, gastric dysrhythmias themselves are potential pathophysiologic mechanisms of FD symptoms and are a therapeutic target. 223 In patients with chronic nausea with normal or delayed gastric emptying treated with aprepitant, nausea symptoms and tachygastria decreased significantly compared with placebo. 224 In other patients with FD, both gastric emptying and GMA are normal. Abnormalities in gastric accommodation, relaxation, or gastric hypersensitivity to distension may account for FD symptoms in these patients. 225-227 Thus, a variety of neuromuscular dysfunctions in different regions of the stomach may be present in patients with FD symptoms. GERD Many patients with typical GERD have additional symptoms of early satiety, postprandial fullness, and nausea, see Chapter 46. Approximately 30% of patients with GERD also have FD. 218 20% to 30% of patients with GERD have delayed gastric emptying. 228-229 Symptoms associated with gastroparesis improved and gastric dysrhythmias reverted to normal. 3 cpm GMA after treatment of the lower esophageal sphincter with radiofrequency ablation, RFA. 230 In some patients, nausea represents a subtle, atypical symptom of esophageal acid reflux and nausea improves after aggressive acid suppression therapy. 231 Lower esophageal sphincter abnormalities, including increased numbers of transient lower esophageal sphincter relaxation, may contribute to inefficient gastric emptying and gastric dysrhythmias reported in these patients. 232-233 GERD patients also tended to retain solids and liquids in the proximal stomach compared with control subjects. 234 An abnormality that may stimulate excessive transient lower esophageal sphincter relaxations. RFA treatment at the lower esophageal sphincter region in patients with heartburn and dyspepsia improved heartburn, gastric emptying rates, and gastric dysrhythmias. 230 These data suggest in some patients with GERD and gastroparesis, treatments of GERD by RFA or fundoplication improves the gastric electrical rhythms and the rate of gastric emptying. Constipation, IBS, and pseudo-obstruction gastroparesis, gastric dysrhythmias, GERD, and IBS are all present in some patients and indicate diffuse or pan-GI neuromuscular dysfunction. 235 Gastroparesis occurs in 60% of patients with constipation predominant IBS. In a study of 350 patients with IGP, 89% were taking proton pump inhibitors and 64% had IBS.214. These patients represent another overlap syndrome of GI neuromuscular disorders. See Chapter 122. Patients with intestinal pseudo-obstruction syndromes often have GERD, gastroparesis, small bowel dysmotility, and colonic inertia and reflect the severest form of generalized GI neuromuscular disorders. 235,236 Pseudo-obstruction secondary to progressive systemic sclerosis, scleroderma, commonly involves the esophagus and stomach but may also cause small bowel dysmotility and subsequent bacterial overgrowth. 237 Intestinal pseudo-obstruction may be due to idiopathic degenerative or inflammatory processes involving the smooth muscle or ends, as discussed in chapters 37 and 124. Pathologic classifications of neuromuscular diseases of the GI tract, neuropathies, myopathies, and ICC abnormalities. Cagulopathies are evolving. 238 A variety of neurologic diseases may also involve the stomach or other regions of the digestive tract and cause pseudo-obstruction-like symptoms. These neurologic disorders include Ehlers-Danlos syndrome. 239 Postural orthostatic tachycardia syndrome. 240 Spinal cord and head injuries, amyotrophic lateral sclerosis, myasthenia gravis, a variety of muscular dystrophies, and Parkinson disease. See Chapter 37. Miscellaneous conditions. Patients with gastroparesis-like symptoms and cirrhosis with portal hypertension. 241 Chronic kidney disease. 242 Chronic pancreatitis. 243 or advanced HIV infections. 244 may have underlying gastroparesis. Patients with the Rett syndrome, which includes Box 501 differential diagnosis of chronic nausea and vomiting, mechanical GI tract obstruction, pylorus, bile duct, small intestine, colon, mucosal inflammation, peritoneal irritation, carcinomas, e.g., gastric, ovarian, renal, bronchogenic, metabolic/endocrine disorders, diabetic mellitus, hypothyroidism, hyperthyroidism, adrenal insufficiency, uremia, medications, anticholinergics, narcotics, L-DOPA, progesterone, calcium channel blockers, digitalis, NSAIDs, antidysrhythmia agents, lobiprostone, cannabis, metformin, amylin analogues, gastroparesis. See Table 50.2, gastric dysrhythmias, tachygastria, bradygastria, mixed, CNS disorders, tumors, migraine, seizures, stroke, orthostatic intolerance, psychogenic disorders, anorexia nervosa, bulimia nervosa. 758 Part 6 Stomach and duodenum. Lack of development, autistic behavior, ataxia, and dementia in young girls, frequently have failure to thrive and significant gastroparesis and esophageal contraction abnormalities. 245 A variety of cancers may have local and systemic effects on the stomach neuromuscular apparatus that result in gastroparesis. 246,247 The neoplastic and perineoplastic neuropathic syndromes and the effects of cytotoxic chemotherapy and radiation may result in gastroparesis and affect the patient's nutrition and intravascular volume status. HP infection does not affect the rate of gastric emptying. On the other hand, abnormal GMA has been reported in patients with HP gastritis, and the dysrhythmias disappeared after eradication of HP. 248 Gastric emptying is delayed in patients with anorexia nervosa. 249 but is normal in patients with bulimia. 250 Cyclic vomiting syndrome is an unusual entity in that days of profound and unremitting nausea and vomiting, that often requires hospitalization, are followed by many days or months with virtually no GI symptoms. 251 Cyclic vomiting syndrome occurs in adults as well as in infants. 251,252 Some patients with orthostatic intolerance present with nausea and symptoms of gastroparesis and reflect autonomic nervous system dysfunction and gastric neuromuscular dysfunction. 240,253 Dumping syndrome and rapid gastric emptying. Dumping syndrome occurs in some patients who have had vagotomy and pyloroplasty or Billroad I or Billroad II gastrojejunostomy. 254 In these patients, the ingested foods are not accommodated and retained in the fundus because of poor fundic relaxation. In addition, the foods are not normally triturated by peristaltic waves if the pylorus has been incised. Thus, liquid and solid nutrients are rapidly emptied or dumped into the duodenum or jejunum. Dumping syndrome can occur in patients who have no history of gastric operations. Thus, dumping syndrome may be overlooked as a diagnosis by many physicians. Symptoms associated with dumping syndrome can mimic the symptoms associated with gastroparesis and FD. The dumping syndrome symptoms include nonspecific abdominal discomfort, bloating, and nausea and vomiting. These symptoms are usually experienced in the first hour after ingestion of foods. Sweating and lightheadedness, however, may occur and be followed by abdominal cramps and diarrhea that occur 2 to 4 hours after the meal and are additional clues to the dumping syndrome. Early symptoms are due to the distension of the small bowel by rapidly emptied solids and liquids from the stomach, whereas symptoms that occur later are due to rapid absorption of carbohydrates and hyperglycemia that is poorly matched in time with secretion of insulin. This mismatch of plasma glucose and insulin can result in symptomatic hypoglycemia. The rapid small bowel transit and poor absorption of the ingested nutrients lead to an osmotic form of diarrhea. 255 Patients with no history of gastric operations may also have rapid gastric emptying, idiopathic dumping syndrome. 254 Including patients with FD. 255 Rapid gastric emptying is present when more than 30% of the test meal, a low-fat egg substitute meal, is emptied in 30 minutes, or more than 70% is emptied at 60 minutes. 38 Rapid gastric emptying is associated with early stages of type 2 diabetes mellitus, see earlier, ZES, and the variety of gastric surgeries described earlier. Idiopathic rapid emptying is diagnosed in patients with no history of gastric operations or other causes. Diagnosis of gastric neuromuscular disorders. History: patients with gastric neuromuscular dysfunction ranging from accommodation defects to gastric dysrhythmias to frank gastroparesis have similar dyspepsia-like symptoms. Most patients with gastric neuromuscular disorders have few upper GI symptoms when they are fasting. However, the ingestion of meals stimulates the disordered gastric neuromuscular apparatus, and early satiety, prolonged epigastric fullness, epigastric discomfort or pain, mild to severe nausea, and vomiting are then experienced. 218 The multiple symptoms are related to neuromuscular disorders of various regions of the stomach, Table 50.4. The symptoms of postprandial fullness, nausea, distension, and vomiting, are symptoms similar to FD, postprandial distress with nausea, or gastroparesis-like symptoms. Patients with IGP have more abdominal pain compared with DGP. 214 Patients with type 2 diabetes mellitus and gastroparesis have more bloating than patients with type 1 diabetes mellitus with gastroparesis. Pain is a dominant symptom in 20% to 30% of patients with IGP. 214,256 Nausea and vomiting are the symptoms that lead to medical evaluation in over 85% of patients with gastroparesis. Vomitus that contains undigested, chewed food is strong evidence for gastroparesis. Prolonged postprandial fullness, weight loss, and female gender are predictive factors for gastroparesis. 257 Bloating is also an important symptom related to gastroparesis but may also be due to small bowel or colonic dysmotility. 258 Patients learn to reduce these postprandial symptoms by adjusting their diet and carry on for months or years before they seek medical attention or their physicians recognize the possibility of gastroparesis. Table 50.4 Disorders of gastric neuromuscular activity, physiology, pathophysiology, and symptoms. Neuromuscular activity. Physiologic effects. Pathophysiologic consequence, S. Pathophysiologic abnormality. Symptoms: receptive relaxation, fundic accommodation, excess or poor accommodation, NNOS, vagotomy, ES, antral peristalsis, trituration, gastric emptying, weak peristalsis, gastroparesis, ICCs, enteric neurons, vagotomy, PF, normal 3 cpm, slow wave, normal peristalsis, gastric emptying, gastric dysrhythmia, e.g., tachygastria, ICCs, vagotomy, nausea, antropyloriduodenal coordination, normal trituration, gastric emptying, pyloraspasm, pyloric stenosis, NNOS, PUD, ES, PF, nausea, vomiting, normal vagus, ANS, CNS function, normal gastric neuromuscular function, fundic dysfunction, gastric dysrhythmias, peristalsis, vagus function, ANS imbalance, ES, PF, nausea, vomiting, ANS, autonomic nervous system, CNS, Central nervous system, CPM, cycles per minute, ES, early satiety, ICCs. Interstitial cells of Cajal, N, nerves, NNOS, neuronal nitric oxide synthetase, PF, prolonged fullness, PUD, peptic ulcer disease. Chapter 50 Gastric Neuromuscular Function and Neuromuscular Disorders 759-50 The gastroparesis cardinal symptom index is used to quantify gastroparesis symptoms. 259 Persistent nausea is one of the most noxious symptoms of the gastric neuromuscular disorders. A thorough review of the causes of nausea and vomiting is required, see Chapter 15, and an appropriate differential diagnosis should be considered, see Box 50.1. Point 76,260 Unexplained nausea and vomiting due to gastric neuromuscular disorders must be distinguished from atypical GERD, regurgitation, and rumination syndrome. Atypical GERD may present as unexplained nausea, because these patients have little or no heartburn. 231 Regurgitation is the gentle delivery of gastric content into the esophagus and pharynx, and the content is sometimes re-swallowed, whereas vomiting is the forceful ejection of gastric contents from the mouth. Rumination refers to the effortless return of ingested liquids and solid foods into the mouth without burning, bitter taste, or nausea. Patients with rumination have impaired gastric accommodation and a more sensitive relaxation of the lower esophageal sphincter pressure in response to gastric distension. 261,262 Rumination occurs in healthy adolescents and young adults but was previously recognized among children with neural and developmental disorders. Abdominal pain, in contrast to the abdominal discomfort of bloating and nausea, occurs in approximately 20% of patients with gastroparesis. 263,264 Any recurrent abdominal pain syndrome in patients must be worked up extensively because the nausea and vomiting may be secondary to the specific cause of the pain, e.g., epigastric pain associated with nausea and vomiting may be due to erosive esophagitis. Once the esophagitis is diagnosed and treated, the pain and nausea disappear. A specific pain syndrome may suggest diagnoses such as cholecystitis, pancreatitis, or SOD. On the other hand, the epigastric discomfort or pain in some gastroparesis patients may originate from the stomach, excessive muscle tone of the fundus, high-amplitude antral contractions, pylorospasm, or hypersensitivity of the stomach are potential causes of pain. 265 Recurrent retching and vomiting often result in abdominal pain, which may be due to abdominal muscle and rib tenderness or the abdominal wall syndrome. 266-268 Physical examination The general examination may be normal or reveal signs of volume depletion, weight loss, and poor nutrition. Orthostatic changes may suggest postural orthostatic tachycardia syndrome. 269 Almost 40% of patients with gastroparesis are overweight or obese. 213,270 Inspection of dentition may show erosion of enamel associated with chronic GERD or bulimia. Abdominal examination may detect masses, organomegaly, and areas of tenderness. Abdominal distension and succession splash are rarely present in gastroparesis. Auscultation over the epigastrium may detect bruits that indicate stenoses of the celiac or superior mesenteric arteries. Pain and tenderness that are precisely localized to healed abdominal incisions and increase when the head is flexed and anterior abdominal muscles are contracted, positive Carnot sign, suggest the pain is from an abdominal wall syndrome and not the stomach. 266-268 Joint laxity may reflect presence of Ehlers-Danlos syndrome. 239 Neurologic examination may reveal nystagmus, facial weakness, ataxia, or other abnormalities, which point to primary neurologic diseases. Standard tests. Common causes of nausea and vomiting and postprandial distress symptoms are excluded by standard endoscopy, routine laboratory studies, and CT of the abdomen and head. 260 Gastric cancers are detected in less than 1% of these patients who undergo endoscopy. 271 If these standard tests are normal and symptoms persist, the gastric neuromuscular disorders should be considered. Non-invasive tests and objective diagnosis of neuromuscular disorders of the stomach can be established by the results of Gets and Egg with water load satiety test. Gets and Egg are complementary in defining different aspects of gastric neuromuscular disorders, see Table 50.3, point 52,132,260 By combining the results of Gets and Egg, 4 pathophysiologic categories of gastric neuromuscular function can be defined and rational treatment approaches designed. The categories are: 1. Gastroparesis with gastric dysrhythmia, 2. Gastroparesis with normal gastric electrical rhythm, 3. Normal gastric emptying with gastric dysrhythmia, and 4. Normal gastric emptying with normal gastric electrical rhythm. The 4 categories provide a conceptual framework for understanding the spectrum of gastric neuromuscular disorders and providing an approach to therapy. See Table 50.2. In regards to patients with gastroparesis, there are 2 subsets. Category 1 patients have severe neuromuscular dysfunction with gastroparesis on GITS and gastric dysrhythmias such as tachygastria. These patients may have diabetic, idiopathic, or post-surgical gastroparesis. In patients with FD, 17% to 32% have both gastric dysrhythmias and gastroparesis. 52,132,136 patients in Category 1 often have more severe symptoms, require many drugs, and may require venting gastrostomies, enteral feeding, and gastric pacing, as discussed later. Category 2 patients have gastroparesis and normal or high-amplitude 3 cpm egg signals. These patients may have fixed mechanical obstructions of the pylorus and duodenum that are reversible with operation. 134 patients with a normal amplitude 3 cpm pattern and gastroparesis more often have normal endoscopy and have pyloric neuromuscular dysfunction that responds to endoscopic therapies. 167 Electromechanical dissociation is also a pathophysiologic possibility. In regards to patients' FD or gastroparesis-like symptoms and normal gastric emptying, there are 2 subtypes. Category 3 patients have symptoms associated with gastroparesis but have normal gastric emptying and gastric dysrhythmias. 52 These patients have gastroparesis-like symptoms. 219 or FDPDS. 218 Category 3 patients had a significantly better response to the prokinetic agent cisapride than patients with normal egg recordings. 220 Category 4 patients have symptoms associated with gastroparesis but normal gastric emptying and normal GMA. The gastroparesis-like symptoms may be due to poor gastric relaxation or gastric visceral hypersensitivity in response to distension with the water load or caloric satiety tests. Diagnoses of non-gastric disorders should also be considered in this patient group. Atypical GERD may cause nausea, and a 24-hour pH study will confirm the relationship. 231 A CCK-stimulated gallbladder emptying study may document gallbladder dysfunction in the absence of cholelithiasis. If postprandial abdominal discomfort/pain and disturbed bowel function are present, then IBS should be considered. CNS diseases and orthostatic intolerance as causes of nausea also should be assessed as outlined in Table 50.1. Treatment: if delayed gastric emptying is confirmed, then the causes of gastroparesis should be reviewed. See Table 50.3. The reversible obstructive causes of gastroparesis due to fixed pyloric stenosis or functional pylorospasm and ischemic gastroparesis due to chronic mesenteric ischemia must be excluded because these entities are reversible. If gastric emptying is normal, then gastric dysrhythmia and gastric accommodation disorders may be the neuromuscular disorders that are relevant to the symptoms. Non-gastric diseases should also be considered. By grouping patients on the basis of GET and egg test results, the pathophysiologic findings can help in the understanding of symptoms and in developing approaches to treatment. See Table 50.2. Treatments listed 760 Part 6 Stomach and duodenum in Tables 50.5 and 50.6 reflect the broad but limited armamentarium that ranges from prokinetic agents to endoscopic and electrical therapies to diet counseling. Drug therapy: prokinetic agents for corpus and antrum drugs with prokinetic effects on gastric contractility and antiarrhythmic effects on gastric dysrhythmias are prescribed for patients in Categories 1 and 3, see Table 50.5. Patients who have gastroparesis and tachygastria have severe electrical and contractile abnormalities of the stomach. The treatment includes prokinetics, antinauseant therapies, and dietary counseling. 164,260 Erythromycin is a macrolide antibiotic and motilin-like molecule that increases gastric emptying by stimulating strong phase 3-like enteral contractions. 272,273 Erythromycin, however, often increases nausea and vomiting symptoms. Metoclopramide, a substituted benzamide related to procainamide, is a useful prokinetic antiemetic but has a black box warning. The drug is approved only in the USA for use for 2 to 8 weeks in gastroparesis, and meticulous follow-up is needed because metoclopramide may cause depression, extrapyramidal side effects, and irreversible tardive dyskinesia. 274,275 Domperidone is a dopamine antagonist that decreases nausea, corrects gastric dysrhythmias, and increases gastric emptying rates. 276,277 Domperidone may be obtained through a compassionate use drug application process with the FDA. Cisapride was not approved for gastric emptying disorders but increased gastric emptying rates and decreased dyspepsia symptoms in some patients. 278 Cisapride was withdrawn from the market but is available through special compassionate use FDA programs. The ghrelin agonist, Relamorelin, stimulated gastric emptying and decreased symptoms in patients with DGP. 279 Pro-relaxant agents for fundus and pylorus. Few drugs relax the fundus. Sumatriptan, a 5-HT1 antagonist, decreases fundic tone but was not better than placebo in reducing symptoms in patients with FD. 280 Buspirone relaxes the fundus and may decrease postprandial fullness. 281 Trials of dicyclamine or calcium channel blockers to decrease fundic tone have not been reported. Botulinum toxin relaxes the pyloric sphincter pressure and is described later in endoscopic therapy. Anti-nausea therapy. Nonspecific approaches to treating nausea and vomiting due to gastric neuromuscular disorders include serotonin, 5-HT3, receptor antagonists such as ondansetron and granisetron, see Table 50.5. These agents, as well as the phenothiazines and antihistamines such as promethazine, dimenhydrinate, and cyclizine, are often used for these symptoms, but there are no controlled trials in patients with gastric neuromuscular disorders. Lorazepam or alprazolam or other anti-anxiety medications reduce nausea in some patients. 175,176,282 Dronabinol and mirtazapine are other drugs used for nausea, but no controlled trial data are available for use in gastroparesis. An uncontrolled trial of tricyclic antidepressants alleviated nausea in approximately 70% of patients with unexplained nausea. 283 But a recent placebo-controlled trial showed that one tricyclic compound, nortriptyline, was no better than placebo in reducing symptoms of gastroparesis. 284 Aprepitant is a neurokinin 1 antagonist that significantly decreased nausea and decreased tachygastria in patients with normal or delayed gastric emptying and chronic nausea. 224 Electrical therapy. Accustimulation, mild electrical stimulation of acupuncture points, reduces nausea of pregnancy, nausea due to chemotherapy agents, postoperative nausea, and the nausea of motion sickness. 285,286 and symptoms related to gastroparesis. 287 Gastric electrical therapies. 3 different methods are being investigated to treat gastroparesis. 1. GES with high frequency, e.g., 12 cpm, and short duration, 300 sec, stimulation. 2. Gastric pacing with low frequency, e.g., 3 cpm, and long duration, 300 millisecond, stimulation, and 3. Sequential neural electrical stimulation with multiple pairs of electrodes positioned on the corpus antrum. Gastric electrical stimulator GES at 12 cpm during a 12-month period of continuous treatment significantly decreased nausea and vomiting in patients with refractory, idiopathic, diabetic, or post-surgical gastroparesis. 288-291 Stimulating the stomach at 12 cpm, 4 times the normal slow-wave frequency, resulted in improvement in nausea and vomiting in patients with gastroparesis. 292 A 12-week placebo-controlled crossover study of GES and DGP showed no benefit, but at 12 months, patients with open-label GES had a significant decrease in symptoms compared with baseline, and gastric emptying rates improved at 1 year of treatment. 293 Uncontrolled studies from several centers indicate that 50% to 70% of patients treated with GES report a decrease in their chronic nausea and vomiting symptoms. Gastroparesis patients with normal 3 cpm egg patterns and more ICCs on gastric biopsies had significantly better symptom response to GES compared with patients with tachygastria and fewer ICCs. 23 Patients with DGP had better symptom responses than patients with IGP. 288,291 Temporary gastric stimulation may help identify GES responders. 293 Intraoperative measurement of gastric slow waves showed tachygastrias, bradygastrias, and aberrant initiation and conduction abnormalities in 11 of 12 patients with gastroparesis, and may help direct pacing and stimulator treatments. 21 Complications of GES include a 10% incidence of infections in the subcutaneous pocket where the device is positioned. There is also a small incidence of fracture of the electrode wires. Gastric pacing. Low-frequency gastric stimulation using a 3 cpm stimulus to pace or entrain the normal slow-wave rhythm in patients with gastroparesis seeks to stimulate 3 gastric peristaltic contractions per minute and improve gastric emptying. 294 In 13 patients, electrical stimulation at a frequency up to 10% higher than the normal 3 cpm, at an amplitude of 4 mA and a pulse width of 300 ms, was used to entrain and pace the slow waves. Gastric dysrhythmias were eradicated in some patients. In a similar study of 9 additional patients, electrical stimulation was used to entrain the slow wave, and tachygastria was converted to normal 3 cpm rhythms in 2 patients. 295 After 1 month of gastric pacing treatment, gastric emptying was significantly improved, symptoms of gastroparesis were significantly reduced. Adverse events due to gastric pacing devices are discomfort at the site of electrical stimulation and fracture or dislodgement of the electrodes. Sequential neural electrical stimulation. Sequential neural electrical gastric stimulation is gastric pacing that uses a microprocessor to sequentially activate a series of electrodes that encircle the distal 2/3 of the stomach. The stimulation sequence induces propagated contractions that cause a forceful emptying of the gastric content. 296 Chapter 50 Gastric Neuromuscular Function and Neuromuscular Disorders 761 Table 50.5 Drug and non-drug therapies used to treat symptoms in patients with gastric neuromuscular disorders. 50 Therapy mechanisms and sites of action. Dose. Adverse effects. Prokinetic drugs. Macrolides. Erythromycin Motilin receptor agonist 125 to 250 mg 4 times daily. Nausea, diarrhea, abdominal cramps, rash. Substituted benzamides. Metoclopramide D2 receptor antagonist, 5-HT3 receptor antagonist, 5-HT4 receptor agonist 5 to 20 mg before meals and at bedtime. Extrapyramidal symptoms, dystonic reactions, anxiety, depression, hyperprolactinemia, tardive dyskinesia. Domperidone D2 receptor antagonist, peripheral, 10 to 20 mg before meals and at bedtime. Hyperprolactinemia, breast tenderness, galactorrhea. Serotonin agonists. Cisapride * 5-HT4 receptor agonist 5 to 20 mg before meals. Cardiac dysrhythmias, diarrhea, abdominal discomfort. Tegaserod * partial 5-HT4 receptor agonist 2 to 6 mg 3 times daily. Diarrhea, abdominal pain. Ghrelin agonist. Relamerlin. Ghrelin agonist to be determined. Glycemic control. Prorelaxa NT drugs. Dicyclamine muscarinic antagonist 10 to 20 mg before meals. Drowsiness, dry mouth. Buspirone 5-HT1 agonist 7.5 to 15 mg 2 times daily. Dizziness, headache, nausea. Botulinum toxin, Botox. See endoscopic therapies. Anti-nausea drugs. Serotonin antagonists. Ondansetron 5. HT3 receptor antagonist 4 to 8 mg 2 to 4 times daily, either orally or for headache. Increased liver enzymes. Granisetron 5-HT3 receptor antagonist 2 mg once daily or 3.1 mg/24 hr. Patch headache. Increased liver enzymes. Phenothiazines. Prochlorperazine CNS 5 to 10 mg 3 times daily. Hypotension, extrapyramidal symptoms. Antihistamines. Promethazine CNS, H1 receptor antagonist 25 mg twice daily. Drowsiness. Dimenhydrinate H1 receptor antagonist 50 mg 4 times daily. Drowsiness. Cyclizine H1 receptor antagonist 50 mg 4 times daily. Drowsiness. Butyrophenones. Droperidol central dopamine receptor antagonist 2.5 to 5 mg for every 2 hr. Sedation, Hypotension antidepressants CNS cites amitriptyline 25 to 100 mg at bedtime. Constipation nortriptyline 10 to 75 mg at bedtime. Constipation mirtazapine 15 mg at bedtime. Weight gain benzodiazepines/cannabinoids CNS lorazepam 0.5 to 1 mg 4 times daily. Drowsiness, lightheadedness alprazolam 0.25 to 0.5 mg 3 times daily. Drowsiness, lightheadedness dronabinol 5 to 10 mg 2 times daily. Sedation substance P agonists aprepotent substance P antagonist 15 mg/day. Tiredness, hiccups. Electrical therapies acupressure and accustomulation/acupuncture. Spinal/vagal afferents. Endorphins variable N/a local tenderness. Gastric electrical stimulation. Vagal afferents 12 cpm 330 millisecond 5 Ma pocket infection. Gastric pacing control. Dysrhythmias, improve gastric emptying 3 cpm 300 sec 4 Ma pocket infection. Endoscopic therapies Botox injection into the pylorus. Relax pyloric muscle 25 to 50 units per quadrant. None continued. 762 Part 6 Stomach and duodenum. Endoscopic therapy. Endoscopic therapies refer to drug or device therapies delivered to the relevant regions of the stomach via endoscopes. The injection of botulinum toxin A into the pylorus to decrease sphincter pressure and to improve gastric emptying and nausea and vomiting in patients with gastroparesis produced results similar to placebo injections. 297-299 However, 33 patients with gastroparesis selected on the basis of normal 3 cpm GMA, functional gastric outlet obstruction, underwent balloon dilation of the pylorus or injection of botulinum toxin and symptoms improved in symptoms in 78% of them. 167 patients with 2 or more successful endoscopic pyloric therapies were offered pyloroplasty for more durable treatment, and all had improved symptoms and 6 had normal gastric emptying. 212 Pyloroplasty for patients undergoing GES improved GE and symptoms. 300 Gastric peroral endoscopic myotomy is an advanced endoscopic procedure during which a pyloromyotomy is performed for the treatment of gastroparesis. Early studies with gastric peroral endoscopic myotomy indicate symptoms and gastric emptying improve after treatment. 301,302 Normal 3 cpm GMA and GP appear to define this subset of patients with functional gastric outlet obstruction due to pylorospasm or dyscalasia. 168,302 Assessment of pyloric distensibility may help further define subgroups. 148 RFA treatment applied to the lower esophageal sphincter area in patients with GERD and dyspepsia improves GERD but also gastric dysrhythmias and gastric emptying. 230 Diet therapy. Dietary counseling. Many patients with acute or chronic nausea and vomiting from gastric neuromuscular disorders do not know what to eat and will benefit from dietary counseling. 260,303 Only one-third of Table 50.5 drug and non-drug therapies used to treat symptoms in patients with gastric neuromuscular disorders. Cont D therapy mechanisms and sites of action. Dose adverse effects. Balloon dilation of the pylorus. Stretch pyloric muscle 20 mm balloon. 2. Min abdominal pain after dilation. Radiofrequency ablation at lay. Reduce gastroesophageal reflux, improve gastric myoelectrical activity and/a transient dysphagia. Diet therapies. Gastroparesis diet based on gastric emptying physiology. See Table 50.6 Non-high protein drinks decreases gastric dysrhythmias. Unknown non-gastrostomy. Venting paretic stomach and/AC Chapter 6 Jejunostomy. Enteral nutritional support and/AC Chapter 6 TPN. Bypass paretic stomach. American Samoa. Needed sepsis, thrombosis of central veins. * Compassionate clearance use only. Botox, botulinum toxin, cpm, cycles per minute, D2, dopamine 2, 5-HT, 5-hydroxytryptamine, H1, histamine 1, N/A, not applicable. Table 50.6 Dietary therapies of nausea and vomiting in patients with gastric neuromuscular disorders. Stepwise approach. Diet goal foods to avoid. Step 1. Sports drinks and bouillon for severe nausea and vomiting, small volumes of salty liquids, with some caloric content to avoid volume depletion. Chewable multiple vitamin 1000 to 1500 ml/day in multiple servings, e.g., 1 to 120 ml servings over 12 to 14 hr. Patient can sip 30 to 60 ml at a time to reach 120 ml/hr. Citrus drinks of all kinds. Highly sweetened drinks. Step 2. Soups and smoothies. If Step 1 is tolerated, soup with noodles or rice and crackers. Smoothies with low-fat dairy, peanut butter, cheese, and crackers in small amounts. Caramels or other chewy confection. Ingest above foods in at least 6 small volume meals/day. Chewable multiple vitamin 1500 calories/day to avoid volume depletion and maintain weight, often more realistic than weight gain. Creamy, milk-based liquids. Step 3. Starches, chicken, fish. If Step 2 is tolerated, noodles, pastas, potatoes, mashed or baked, rice, baked chicken breast, fish, all easily mixed and emptied by the stomach. Ingest solids in at least 6 small volume meals/day. Multiple vitamin, liquid or dissolvable, common foods that the patient finds interesting and satisfying and that provoke minimal nausea/vomiting symptoms. Fatty foods that delay gastric emptying, red meats and fresh vegetables that require considerable trituration, pulpy fibrous foods that promote formation of bezoars. From Cook K.L., Hassler W.L., editors. Nausea and vomiting, diagnosis and treatment. Switzerland, Springer International Publishing, 2017. Chapter 50. Gastric neuromuscular function and neuromuscular disorders. 763. 50 patients with gastroparesis have had a dietary consultation. Point 269. In the nausea/vomiting, gastroparesis, diet, liquid and solid foods that are easy for the stomach to mix and empty are prescribed. Point 260,303. This diet is based on gastric emptying principles and is a 3-step diet that requires minimal neuromuscular work of the stomach as the diet is advanced. See Table 50.6. Step 1 is primarily electrolyte solutions that are consumed in small amounts over a 24-hour period in order to avoid volume depletion. Liquids require less gastric neuromuscular work to empty than solid foods. If patients tolerate Step 1, then Step 2 may be tried next. Step 2 diet includes soups containing noodles or rice. Milk-based creamy soups are avoided. Step 3 emphasizes starches and chicken and turkey breast. These solid foods require less gastric work to mix and empty compared with fresh vegetables or red meats. Fried and greasy foods are avoided because fats delay gastric emptying. A chewable multiple vitamin is taken daily in all 3 steps. Nutraceuticals. Liquid protein meals with or without ginger decrease nausea associated with motion sickness and gastric dysrhythmias, nausea of pregnancy, and delayed nausea after chemotherapy. Point 304-308. These protein-based meal therapies have not been formally evaluated in patients with gastroparesis or gastric dysrhythmias. Other approaches for patients with chronic nausea and vomiting: percutaneous endoscopic gastrostomy, PEG, tubes may be placed for periodic venting gastric contents to avoid frequent vomiting episodes and to improve quality of life. Point 309. The venting PEG does not treat the underlying gastric neuromuscular disorder, but it allows the patients to empty the stomach rather than suffer repeated episodes of emesis and discomfort. Hospitalizations are reduced. Point 175. Medications and some nutritional liquids may be tolerated when given through the gastrostomy tube. Jejunal feeding tubes for enteral nutrition may be needed to provide basic caloric support for patients with severe nausea and vomiting from gastric neuromuscular disorders. A PEG with J-tube extension usually fails because a single vomiting episode may propel the extension tube into the stomach. A J-tube placed endoscopically or surgically is required. See Chapter 6. Point 309. TPN via central 4 catheters should be avoided if at all possible because of the frequent development of line sepsis and occasional venous thrombosis.